17 Gene expression, from gene to protein. Figure 17.1. 4 albino donkeys with 1 brown donkey. A population of albino donkeys grazes on vegetation on the hillsides of Asinara, an Italian island. Several centuries ago, a recessive mutation that disables pigment synthesis arose in the DNA of one donkey and was passed down through the generations. Inbreeding has resulted in a large number of homozygous albino donkeys living on the island today. How can one change in DNA result in such a dramatic change in appearance? 17.1 to 1 full alternative text. Key concepts. 17.1 Genes specify proteins via transcription and translation. 336. 17.2 Transcription is the DNA-directed synthesis of RNA, a closer loop. 342. 17.3 Eukaryotic cells modify RNA after transcription. 345. 17.4 Translation is the RNA-directed synthesis of a polypeptide, a closer loop. 347. 17.5 Mutations of one or a few nucleotides can affect protein structure and function. 357. Study tip. Make a visual study guide, sketch the process shown below, and add labels and details as you read the chapter. In this exercise, assume all processes take place in a eukaryotic cell. The process of transcription and translation is partially drawn. Go to Mastering Biology. For students, in eText and Study area. Get ready for Chapter 17. Bioflix Registered Animation, Protein Synthesis. Figure 17.27 Walkthrough, Types of Small-Scale Mutations that Affect mRNA Sequence. For instructors to assign, in Item Library. Bioflix Registered Tutorial, Protein Synthesis, 1 of 3 Overview. Tutorial, CRISPR, A Revolution in Genome Editing. Ready to Go Teaching Module, in Instructor Resources. Gene Expression, Mutations. Concept 17.5. Concept 17.1 Genes specify proteins via transcription and translation. Inherited traits like albinism are determined by genes, which have information content in the form of specific sequences of nucleotides along stretches of DNA, the genetic material. The DNA inherited by an organism leads to specific traits by dictating the synthesis of proteins and of RNA molecules involved in protein synthesis. In the case of coat and skin color, the gene containing the information to synthesize an enzyme that makes pigment allows normal coloring, and a faulty version of that gene leads to lack of color, or the albino phenotype. Proteins are the link between genotype and phenotype. Gene expression is the process by which DNA directs the synthesis of proteins, or, in some cases, just RNAs. The expression of genes that code for proteins includes 2 stages, transcription and translation. Before going into the details of how genes direct protein synthesis, let's step back and examine how the fundamental relationship between genes and proteins was discovered. Evidence from studying metabolic defects. In 1902, British physician Archibald Garrod was the first to suggest that genes dictate phenotypes through enzymes, proteins that catalyze specific chemical reactions in the cell. He postulated that the symptoms of an inherited disease reflect an inability to make a particular enzyme. He later referred to such diseases as inborn errors of metabolism. For example, people with a disease called alkaptonuria have black urine because it contains a chemical called alkapton, which darkens upon exposure to air. Garrod reasoned that these people cannot make an enzyme that breaks down alkapton, so alkapton is expelled in their urine. Several decades later, research supported Garrod's hypothesis that a gene dictates the production of a specific enzyme, later named the one gene, one enzyme hypothesis. Biochemists learned that cells synthesize and degrade most organic molecules via metabolic pathways, in which each chemical reaction in a sequence is catalyzed by a specific enzyme. See Concept 8.1. Such metabolic pathways lead, for instance, to the synthesis of the pigments that give the brown donkey in Figure 17.1. It's for color or fruit flies, Drosophila, their eye color, see. Figure 15.3. In the 1930s, the American biochemist and geneticist George Beadle and his French colleague Boris F. Russy speculated that in Drosophila, each mutation affecting eye color blocks pigment synthesis at a specific step by preventing production of the enzyme that catalyzes that step. But neither the chemical reactions nor the enzymes that catalyze them were known at the time. Nutritional mutants in Neurospora, scientific inquiry. A breakthrough in demonstrating the relationship between genes and enzymes came a few years later at Stanford University, where Beadle and Edward Tatum began working with a bread mold, Neurospora crassa. Unlike the diploid organisms studied by Mendel, Pease, and T.H. Morgan, fruit flies, Neurospora is a haploid species. Because its genome contains only one copy of each gene, that single copy determines the individual's expressed phenotype. Therefore, when Beadle and Tatum wanted to discover the function of any gene, they only needed to mutate and disable that one allele. They chose to study the protein-coding genes required for a specific nutritional activity. They caused mutations in genes by bombarding Neurospora with X-rays and looked among the survivors for mutants that differed in their nutritional needs from the wild-type bread mold. Wild-type Neurospora has modest food requirements. It can grow in the laboratory on minimal medium, a simple solution containing minimal nutrients for growth in organic salts, glucose, and the vitamin biotin incorporated into agar, a support medium. From these basics, wild-type mold cells use their metabolic pathways to produce all the other nutrient molecules, like amino acids, they need for growth, dividing repeatedly and forming visible colonies of genetically identical cells. As shown in Figure 17.2, Beadle and Tatum generated different nutritional mutants of Neurospora cells, each of which had a mutation in one gene and was unable to synthesize a particular essential nutrient. Such cells could not grow on minimal medium but could grow on complete medium, which contains all nutrients needed for growth. For Neurospora, the complete medium consists of the minimal medium supplemented with all 20 amino acids and a few other nutrients. Beadle and Tatum hypothesized that in each nutritional mutant, the gene for the enzyme that synthesizes a particular nutrient had been disabled by the mutation. They were able to determine experimentally which nutrient each mutant strain, original cell and its descendants, was unable to synthesize. Figure 17.2. Beadle and Tatum's experimental approach presents Beadle and Tatum's experimental approach in 6 steps. To obtain nutritional mutants, Beadle and Tatum exposed Neurospora cells to X-rays, inducing mutations, then screened for, tested for, mutants that had new nutritional requirements, such as arginine, as shown here. Figure 17.2. Full alternative text. This approach resulted in a valuable collection of mutant strains of Neurospora, each catalogued by its defect in a particular pathway. 2 colleagues, Adrian S.R.B. and Norman Horowitz, used a collection of arginine-requiring mutants to investigate the biochemical pathway for arginine synthesis in Neurospora. Figure 17.3. SRB and Horowitz pinned down each mutant's defect more specifically, using additional tests to distinguish among 3 classes of arginine-requiring mutants. Mutants in each class required a different set of compounds along the arginine synthesizing pathway, which has 3 steps. These results, and those of many similar experiments done by Beadle and Tatum, suggested that each class was blocked at a different step in this pathway because mutants in that class lacked the enzyme that catalyzes the block step. Figure 17.3. Inquiry. Do individual genes specify the enzymes that function in a biochemical pathway? A. Postulated arginine synthesizing pathway. Precursor with enzyme A produces ornithine. Ornithine with enzyme B produces citrulline. Citrulline with enzyme C produces arginine. Experiment working with the mold Neurospora crassa, Adrian S.R.B. and Norman Horowitz, then at Stanford University, used Beadle and Tatum's experimental approach, see. Figure 17.2, to isolate mutants that required arginine in their growth medium. The researchers showed that these mutants fell into 3 classes, each defective in a different gene. From studies by others on mammalian liver cells, they suspected that the metabolic pathway of arginine biosynthesis involved a precursor nutrient and the intermediate molecules ornithine and citrulline, as shown in the diagram on the right. Their most famous experiment, shown here, tested both the one gene, one enzyme hypothesis and their postulated arginine synthesizing pathway. In this experiment, they grew their 3 classes of mutants under the 4 different conditions shown in the results table below. They included minimal medium, MM, as a control, knowing that wild-type cells could grow on MM but mutant cells could not. See test tubes below. 2 test tubes, 1, growth, wild-type cells growing and dividing, 2, no growth, mutant cells cannot grow and divide. Both contain minimal medium. Control, minimal medium. A results table. 17.125 Full alternative text. Results as shown in the table on the right. The wild-type strain was capable of growth under all experimental conditions, requiring only the minimal medium. The three classes of mutants each had a specific set of growth requirements. For example, Class II mutants could not grow when ornithine alone was added but could grow when either citrulline or arginine was added. A results table. 17.126 Full alternative text. Conclusion from the growth requirements of the mutants, SRB and Horowitz deduced that each class of mutant was unable to carry out one step in the pathway for synthesizing arginine, presumably because it lacked the necessary enzyme, as shown in the table on the right. Because each of their mutants was mutated in a single gene, they concluded that each mutated gene must normally dictate the production of one enzyme. Their results supported the one gene, one enzyme hypothesis, proposed by Beadle and Tatum, and also confirmed that the arginine pathway described in the mammalian liver also operates in Neurospora. Notice in the results table that a mutant can grow only if supplied with a compound made after the defective step because this bypasses the defect. Data from AMSRB and NH Horowitz, the ornithine cycle in Neurospora and its genetic control, Journal of Biological Chemistry 154:129, 139, 1944. What if? Suppose the experiment had shown that Class I mutants could grow only in MM supplemented by ornithine or arginine and that Class II mutants could grow in MM supplemented by citrulline, ornithine, or arginine. What conclusions would the researchers have drawn from those results regarding the biochemical pathway and the defect in Class I and Class II mutants? Because Beadle and Tatum set up their experimental conditions so that each mutant was defective in a single gene, the collected results, taken together, provided strong support for the one gene, one enzyme hypothesis, as they dubbed it, that the function of a gene is to dictate the production of a specific enzyme. Further support for this hypothesis came from experiments that identified the specific enzymes lacking in the mutants. Beadle and Tatum shared a Nobel Prize in 1958 for their discovery that genes act by regulating definite chemical events, in the words of the Nobel Committee. Today, we know of countless examples in which a mutation in a gene causes a faulty enzyme that in turn leads to an identifiable condition. The albino donkeys in Figure 17.1 lack a key enzyme called tyrosinase in the metabolic pathway that produces melanin, a dark pigment. The absence of melanin causes white fur and other effects throughout the donkey's body. Its nose, ears, and hooves, as well as its eyes, are pink because no melanin is present to mask the reddish color of the blood vessels that run through those structures. The products of gene expression, a developing story. As researchers learned more about proteins, they made revisions to the one gene, one enzyme hypothesis. First of all, not all proteins are enzymes. Keratin, the structural protein of animal hair, and the hormone insulin are two examples of non-enzyme proteins. Because proteins that are not enzymes are nevertheless gene products, molecular biologists began to think in terms of one gene, one protein. However, many proteins are constructed from two or more different polypeptide chains, and each polypeptide is specified by its own gene. For example, hemoglobin, the oxygen-transporting protein of vertebrate red blood cells, contains two kinds of polypeptides, and thus two genes code for this protein, one for each type of polypeptide. See Figure 5.18. Beadle and Tatum's idea was therefore restated as the one gene, one polypeptide hypothesis. Even this description is not entirely accurate, though. First, in many cases, a eukaryotic gene can code for a set of closely related polypeptides via a process called alternative splicing, which you will learn about later in this chapter. Second, quite a few genes code for RNA molecules that have important functions in cells even though they are never translated into protein. For now, we will focus on genes that do code for polypeptides. Note that it is common to refer to these gene products as proteins, a practice you will encounter in this book rather than more precisely as polypeptides. Basic Principles of transcription and translation. Genes provide the instructions for making specific proteins, but a gene does not build a protein directly. The bridge between DNA and protein synthesis is the nucleic acid RNA. RNA is chemically similar to DNA except that it contains ribose instead of deoxyribose as its sugar and has the nitrogenous base uracil rather than thymine, see Figure 5.23. Thus, each nucleotide along a DNA strand has A, G, C, or T as its base, and each nucleotide along an RNA strand has A, G, C, or U as its base. An RNA molecule usually consists of a single strand. We often describe the flow of information from gene to protein in terms of languages. Just as specific sequences of letters communicate information in a language such as English, specific sequences of monomers convey information in polymers like nucleic acids and proteins. In DNA or RNA, the monomers are the 4 types of nucleotides listed previously, which differ in their nitrogenous bases. Genes are typically hundreds or thousands of nucleotides long, each gene having a specific sequence of nucleotides. Each polypeptide of a protein also has monomers arranged in a particular linear order, the protein's primary structure, see Figure 5.18, but its monomers are amino acids. Thus, nucleic acids and proteins contain information written in 2 different chemical languages. Getting from DNA to protein involves 2 major stages, transcription and translation. Transcription is the synthesis, production, of RNA using information in the DNA. The 2 nucleic acids are written in different forms of the same language, and the information is simply transcribed, or rewritten, from DNA to RNA. Just as a DNA strand provides a template for making a new complementary strand during DNA replication, see Concept 16.2, it also can serve as a template for assembling a complementary sequence of RNA nucleotides. For a protein-coding gene, the resulting RNA molecule is a faithful transcript of the gene's protein-building instructions. This type of RNA molecule is called messenger RNA, mRNA, because it carries a genetic message from the DNA to the protein synthesizing machinery of the cell. Transcription is the general term for the synthesis of any kind of RNA on a DNA template. Later, you will learn about some other types of RNA produced by transcription. Translation is the synthesis of a polypeptide using the information in the mRNA. During this stage, there is a change in language. The cell must translate the nucleotide sequence of an mRNA molecule into the amino acid sequence of a polypeptide. The sites of translation are ribosomes, molecular complexes that facilitate the orderly linking of amino acids into polypeptide chains. Transcription and translation occur in all organisms. Because most studies have involved bacteria and eukaryotic cells, they are our main focus in this chapter. Our understanding of transcription and translation in archaea lags behind, but we do know that archaeal cells share some features of gene expression with bacteria and others with eukaryotes. The basic mechanics of transcription and translation are similar for bacteria and eukaryotes, but there is an important difference in the flow of genetic information within the cells. Bacteria do not have nuclei. Therefore, nuclear membranes do not separate bacterial DNA and mRNA from ribosomes and the other protein synthesizing equipment. Figure 17.4a. This lack of compartmentalization allows translation of an mRNA to begin while its transcription is still in progress, as you'll see later. By contrast, eukaryotic cells have nuclei. The presence of a nuclear envelope separates transcription from translation in space and time. Figure 17.4b. Transcription occurs in the nucleus, but the mRNA must be transported to the cytoplasm for translation. In eukaryotes, before RNA transcripts from protein-coding genes can leave the nucleus, they are modified in various ways to produce the final, functional mRNA. The transcription of a protein-coding eukaryotic gene results in pre-mRNA, and further RNA processing yields the finished mRNA. The initial RNA transcript from any gene, including those specifying RNA that is not translated into protein, such as tRNA and rRNA, which will be described in Concept 17.4, is more generally called a primary transcript. Figure 17.4 Overview, the roles of transcription and translation in the flow of genetic information. The roles of transcription and translation in the flow of genetic information. In a cell, inherited information flows from DNA to RNA to protein. The 2 main stages of information flow are transcription and translation. A miniature version of part A, or B, accompanies several figures later in the chapter as an orientation diagram to help you see where a particular figure fits into the overall scheme of gene expression. Figure 17.4 Full alternative text. Mastering biology. Animation, Overview of protein synthesis in bacteria. Animation, overview of protein synthesis in eukaryotes. To summarize, genes program protein synthesis via genetic messages in the form of messenger RNA. Put another way, cells are governed by a molecular chain of command with a directional flow of genetic information. DNA flows to RNA flows to protein. This idea, that the flow of information went only one way, was named the central dogma by Francis Crick in 1956. In the 1970s, however, scientists were surprised to discover some enzymes that use RNA molecules as templates for DNA synthesis, an example of information flow from RNA to DNA, see Concept 19.2. Still, these exceptions do not discredit the idea that, in general, genetic information flows from DNA to RNA to protein. In the next section, we discuss how the instructions for assembling amino acids into a specific order are encoded in nucleic acids. The genetic code. When biologists began to suspect that the instructions for protein synthesis were encoded in DNA, they recognized a problem: there are only 4 nucleotide bases to specify 20 amino acids. Thus, the genetic code cannot be a language like Chinese, where each written symbol corresponds to a word. How many nucleotides, then, would turn out to correspond to an amino acid? Codons, triplets of nucleotides. If each kind of nucleotide base were translated into an amino acid, only 4 amino acids could be specified, 1 per nucleotide base. Would a language of 2-letter code words suffice? The 2-nucleotide sequence AG, for example, could specify 1 amino acid, and GT could specify another. Since there are 4 possible nucleotide bases in each position, this would give us 16, that is, possible arrangements still not enough to code for all 20 amino acids. Triplets of nucleotide bases are the smallest units of uniform length that can code for all the amino acids. If each arrangement of 3 consecutive nucleotide bases specifies an amino acid, there can be 64, that is, possible code words more than enough to specify all the amino acids. Experiments have verified that the flow of information from gene to protein is based on a triplet code. The genetic instructions for a polypeptide chain are written in the DNA as a series of non-overlapping, 3-nucleotide words. The series of words in a gene is transcribed into a complementary series of non-overlapping, 3-nucleotide words in mRNA, which is then translated into a chain of amino acids. Figure 17.5. Figure 17.5. The triplet code. Ribbon model of DNA with 3 regions labeled, genes 1, 2, and 3. Arrows zoom in on a portion of gene 3 to illustrate transcription and translation from DNA template. For each gene, 1 DNA strand functions as a template for transcription of RNAs, such as mRNA. The base pairing rules for DNA synthesis also guide transcription, except that RNA is made with uracil, U, instead of thymine, T. During translation, the mRNA is read as a sequence of nucleotide triplets, called codons. Each codon specifies an amino acid to be added to the growing polypeptide chain. The mRNA is read in the direction. Figure 17.5. Full alternative text. Visual skills. By convention, the non-template strand, also called the coding strand, is used to represent a DNA sequence. Write the sequence of the mRNA strand and the non-template strand in both cases, reading from to and compare them. Why do you think this convention was adopted? Hint: why is this called the coding strand? Mastering biology. MP3 tutor, DNA to RNA to protein. During transcription, the gene determines the sequence of nucleotide bases along the length of the RNA molecule that is being synthesized. For each gene, only one of the 2 DNA strands is transcribed. This strand is called the template strand because it provides the pattern, or template, for the sequence of nucleotides in an RNA transcript. For any given gene, the same strand is used as the template every time that gene is transcribed. However, farther along on the same chromosomal DNA molecule, the opposite strand may function as the template for a different gene. Particular DNA sequences associated with a gene determine how the enzyme that transcribes genes is oriented when it binds, and this establishes which strand will be used as the template. An mRNA molecule is complementary rather than identical to its DNA template because RNA nucleotides are assembled on the template according to base pairing rules, see Figure 17.5. The pairs are similar to those that form during DNA replication, except that U, the RNA substitute for T, pairs with A and the mRNA nucleotides contain ribose instead of deoxyribose. Like a new strand of DNA, the RNA molecule is synthesized in an anti-parallel direction to the template strand of DNA. To review what is meant by antiparallel and the N ends of a nucleic acid chain, see Figure 16.7. In the example in Figure 17.5, the nucleotide triplet ACC along the DNA template strand, written as, provides a template for in the mRNA molecule. The mRNA nucleotide triplets are called codons, and they are customarily written in the direction. In our example, UG is the codon for the amino acid tryptophan, abbreviated TRP, or W. The term codon is also used for the DNA nucleotide triplets along the non-template strand. These codons are complementary to the template strand and thus identical in sequence to the mRNA, except for Ts instead of Us. For this reason, the non-template DNA strand is often called the coding strand. By convention, the sequence of the coding strand is used when a gene sequence is reported. During translation, the sequence of codons along an mRNA molecule is decoded, or translated, into a sequence of amino acids making up a polypeptide chain. The codons are read by the translation machinery in the direction along the mRNA. Each codon specifies which one of the 20 amino acids will be incorporated at the corresponding position along a polypeptide. Because codons are nucleotide triplets, the number of nucleotides making up a genetic message must be 3 times the number of amino acids in the protein product. For example, 300 nucleotides along an mRNA strand code for 100 amino acids in the polypeptide it encodes. Cracking the code. Molecular biologists cracked the genetic code of life in the early 1960s when a series of elegant experiments disclosed the amino acid translations of each of the mRNA codons. The first codon was deciphered in 1961 by Marshall Nierenberg, of the National Institutes of Health, and colleagues. They synthesized an artificial mRNA made up of only uracil-bearing nucleotides, thus containing only one codon, UUU, over and over. When added to a test tube mixture that contained all 20 amino acids, ribosomes, and the other components required for protein synthesis, the poly-U mRNA was translated into a polypeptide containing many units of the phenylalanine, PHE, or F, amino acids, strung together as a polyphenylalanine chain. Thus, Nierenberg determined that the mRNA codon UUU specifies the amino acid phenylalanine. Soon, the amino acids specified by the codons AAA, GGG, and CCC were similarly identified. Although more elaborate techniques were required to decode mixed triplets such as AUA and CGA, all 64 codons were deciphered by the mid-1960s. As Figure 17.6 shows, 61 of the 64 triplets code for amino acids. The other 3 codons act as stop signals, or termination codons, marking the end of translation. Notice that the codon OG has a dual function. It codes for the amino acid methionine, MET, or M, and also functions as a start signal, or initiation codon. Genetic messages usually begin with the mRNA codon OG, which signals the protein synthesizing machinery to begin translating the mRNA at that location. Because OG also stands for methionine, polypeptide chains all begin with methionine when they are synthesized. However, an enzyme may subsequently remove the starter amino acid from the chain. Figure 17.6 The codon table for mRNA. Codon table. The 3 nucleotide bases of an mRNA codon are designated here as the first, second, and third bases, reading in the direction along the mRNA. The codon OG not only stands for the amino acid methionine, MET, or M, but also functions as a start signal for ribosomes to begin translating the mRNA at that point. 3 of the 64 codons function as stop signals, marking where ribosomes end translation. Both 1 and 3 letter codes are shown for the amino acids, see Figure 5.14 for their full names. Figure 17.6 Full alternative text. Visual skills. A segment in the middle of an mRNA has the sequence AGAGA-. Using the codon table, translate the sequence, assuming the first 3 nucleotides are a codon. Mastering biology. Animation, translation, the genetic code. Notice in Figure 17.6 that there is redundancy in the genetic code, but no ambiguity. For example, although codons GAA and GAG both specify glutamic acid, redundancy, neither of them ever specifies any other amino acid, no ambiguity. The redundancy in the code is not altogether random. In many cases, codons that are synonyms for a particular amino acid differ only in the third nucleotide base of the triplet. We will see why this is later in the chapter. Our ability to extract the intended message from a written language depends on reading the symbols in the correct groupings, that is, in the correct reading frame. Consider the statement, the red dog ate the bug. Group the letters incorrectly by starting at the wrong point, and the result will probably be gibberish, for example, her ed oga ted hebuji. The reading frame is also important in the molecular language of cells. The short stretch of polypeptide shown in Figure 17.5, for instance, will be made correctly only if the mRNA nucleotides are read from left to right, in the groups of 3 shown in the figure, uguumkuka. Although a genetic message is written with no spaces between the codons, the cell's protein synthesizing machinery reads the message as a series of non-overlapping 3-letter words. The message is not read as a series of overlapping words uguo, and so on, which would convey a very different message. Evolution of the genetic code. Evolution. The genetic code is nearly universal, shared by organisms from the simplest bacteria to the most complex plants and animals. The mRNA codon CCG, for instance, is translated as the amino acid proline in all organisms whose genetic code has been examined. In laboratory experiments, genes can be transcribed and translated after being transplanted from one species to another, sometimes with quite striking results, as shown in Figure 17.7. Bacteria can be programmed by the insertion of human genes to synthesize certain human proteins for medical use, such as insulin. Such applications have produced many exciting developments in the area of biotechnology. See Concept 20.4. Figure 17.7. Evidence for evolution, expression of genes from different species. Expression of genes from different species. Because diverse forms of life share a common genetic code due to their shared ancestry, one species can be programmed to produce proteins characteristic of a second species by introducing DNA from the second species into the first. Figure 17.7. Full alternative text. Mastering biology. Video, GFP transgenic mice. The evolutionary significance of the code's near universality is clear. A language shared by all living things must have been operating very early in the history of life, early enough to be present in the common ancestor of all present-day organisms. A shared genetic vocabulary is a reminder of the kinship of all life. Concept check 17.1. Make connections. In a research article about alkaptonuria published in 1902, Garrod suggested that humans inherit 2 characters, alleles, for a particular enzyme, and that both parents must contribute a faulty version for the offspring to have alkaptonuria. Today, would this disorder be called dominant or recessive? See Concept 14.4. Describe the polypeptide product you would expect from a poly-G mRNA that is 30 nucleotides long. Draw it. The template strand of a gene contains the sequenced tag dash. Suppose that the non-template sequence was transcribed instead of the template sequence. Draw the non-template sequence into order. Then draw the mRNA sequence and translate it using Figure 17.6. Predict how well the protein synthesized from the non-template strand would function, if at all. For suggested answers, see Appendix A. Concept 17.2 Transcription is the DNA-directed synthesis of RNA, a closer look. Now that we have considered the linguistic logic and evolutionary significance of the genetic code, we are ready to re-examine transcription, the first stage of gene expression, in greater detail. Molecular components of transcription. Messenger RNA, the carrier of information from DNA to the cell's protein synthesizing machinery, is transcribed from the template strand of a gene. An enzyme called an RNA polymerase pries the two strands of DNA apart and joins together RNA nucleotides complementary to the DNA template strand, thus elongating the RNA polynucleotide. Figure 17.8. Like the DNA polymerases that function in DNA replication, RNA polymerases can assemble a polynucleotide only in its direction, adding onto its end. Unlike DNA polymerases, however, RNA polymerases are able to start a chain from scratch. They don't need to add the first nucleotide onto a pre-existing primer. Figure 17.8. The stages of transcription: initiation, elongation, and termination. The stages of transcription. This general depiction of transcription applies to both bacteria and eukaryotes, but the details of termination differ, as described in the text. Also, in a bacterium, the RNA transcript is immediately usable as mRNA. In a eukaryote, the RNA transcript must first undergo processing. Figure 17.8. Full alternative text. Make connections. Compare the use of a template strand during transcription and replication. See Figure 16.18. Mastering biology. Animation, overview of transcription. Animation, overview of transcription in bacteria. Specific sequences of nucleotides along the DNA mark where transcription of a gene begins and ends. The DNA sequence where RNA polymerase attaches and initiates transcription is known as the promoter. In bacteria, the sequence that signals the end of transcription is called the terminator. The termination mechanism in eukaryotes will be described later. Molecular biologists refer to the direction of transcription as downstream and the other direction as upstream. These terms are also used to describe the positions of nucleotide sequences within the DNA or RNA. Thus, the promoter sequence in DNA is said to be upstream from the terminator. The stretch of DNA downstream from the promoter that is transcribed into an RNA molecule is called a transcription unit. Bacteria have a single type of RNA polymerase that synthesizes not only mRNA but also other types of RNA that function in gene expression, such as ribosomal RNA. In contrast, eukaryotes have at least 3 types of RNA polymerase in their nuclei. The one used for pre-mRNA synthesis is called RNA polymerase II. The other RNA polymerases transcribe RNA molecules that are not translated into protein. In the discussion that follows, we start with the features of mRNA synthesis common to both bacteria and eukaryotes and then describe some key differences. Synthesis of an RNA transcript. The 3 stages of transcription, as shown in Figure 17.8 and described next, are initiation, elongation, and termination of the RNA chain. Study Figure 17.8 to familiarize yourself with the stages and the terms used to describe them. RNA polymerase binding and initiation of transcription. The promoter sequence of a gene includes within it the transcription start point, the nucleotide where RNA polymerase actually begins synthesizing the mRNA and typically extends several dozen or so nucleotide pairs upstream from the start point. Figure 17.9. Based on interactions with proteins, transcription factors, RNA polymerase binds in a precise location and orientation on the promoter. This binding determines where transcription starts and the direction it will travel, thus which strand of DNA is used as the template. Figure 17.9. The initiation of transcription at a eukaryotic promoter. The initiation of transcription at a eukaryotic promoter. In eukaryotic cells, proteins called transcription factors mediate the initiation of transcription by RNA polymerase II. Figure 17.9. Full alternative text. Explain how the interaction of RNA polymerase with the promoter would differ if the figure showed transcription initiation for bacteria. Certain sections of a promoter sequence are especially important for binding RNA polymerase in a way that ensures that transcription will begin at the correct start point. In bacteria, part of the RNA polymerase itself specifically recognizes and binds to the promoter. In eukaryotes, a collection of proteins called transcription factors, purple proteins in Figure 17.9, help guide the binding of RNA polymerase and the initiation of transcription. Only after transcription factors are attached to the promoter does RNA polymerase II bind to it. The whole complex of transcription factors and RNA polymerase II bound to the promoter is called a transcription initiation complex. Figure 17.9 shows the role of transcription factors and a crucial promoter DNA sequence called the TATA box informing the initiation complex at a eukaryotic promoter. The interaction between eukaryotic RNA polymerase II and transcription factors is an example of the importance of protein-protein interactions in controlling eukaryotic transcription. Once the appropriate transcription factors are firmly attached to the promoter DNA and the polymerase is bound to them in the correct orientation on the DNA, the enzyme unwinds the two DNA strands and begins transcribing the template strand at the start point. Elongation of the RNA strand. As RNA polymerase moves along the DNA, it untwists the double helix, exposing about 10 to 20 DNA nucleotides at a time for pairing with RNA nucleotides. Figure 17.10. The enzyme adds nucleotides to the end of the growing RNA molecule as it moves along the double helix. As transcription proceeds forward, the newly synthesized RNA molecule behind the RNA polymerase peels away from its DNA template, and the DNA double helix reforms. Transcription progresses at a rate of about 40 nucleotides per second in eukaryotes. Figure 17.10 Transcription elongation. Unwound section of DNA within RNA polymerase. RNA nucleotides added to 3' end of RNA on template DNA strand, shown 3' to 5' left to right. RNA polymerase moves along the DNA template strand, joining complementary RNA nucleotides to the end of the growing RNA transcript. Behind the polymerase, the new RNA peels away from the template strand, which reforms a double helix with the non-template strand. Figure 17.10 Full alternative text. Mastering biology. Bioflix registered animation, transcription. Animation, elongation of the RNA strand. A single gene can be transcribed simultaneously by several molecules of RNA polymerase following each other like trucks in a convoy. A growing strand of RNA trails off from each polymerase, with the length of each new strand reflecting how far along the template the enzyme has traveled from the start point. See the mRNA molecules in Figure 17.23. The congregation of many polymerase molecules simultaneously transcribing a single gene increases the amount of mRNA transcribed from it, which helps the cell make the encoded protein in large amounts. Termination of transcription. Bacteria and eukaryotes differ in the way they terminate transcription. In bacteria, transcription proceeds through a terminator sequence in the DNA. The transcribed terminator, an RNA sequence, functions as the termination signal, causing the polymerase to detach from the DNA and release the transcript, which requires no further modification before translation. In eukaryotes, RNA polymerase II transcribes a sequence on the DNA called the polyadenylation signal sequence, which specifies a polyadenylation signal, AAUAAA, in the pre-mRNA. This is called a signal because once the stretch of 6 RNA nucleotides appears, it is immediately bound by certain proteins in the nucleus. Then, at a point about 10 to 35 nucleotides downstream from the AAUAAA, these proteins cut the RNA transcript free from the polymerase, releasing the pre-mRNA. The pre-mRNA then undergoes processing, the topic of the next section. Although that cleavage marks the end of the mRNA, the RNA polymerase II continues to transcribe. Enzymes begin to degrade the RNA made after cleavage, starting at its newly exposed end. The polymerase continues transcribing, pursued by the enzymes, until they catch up to the polymerase and it dissociates from the DNA. Concept check 17.2. What is a promoter? Is it located at the upstream or downstream end of a transcription unit? What enables RNA polymerase to start transcribing a gene at the right place on the DNA in a bacterial cell? In a eukaryotic cell? What if? Suppose X-rays caused a sequence change in the TATA box of a particular gene's promoter. How would that affect transcription of the gene? See Figure 17.9. For suggested answers, see Appendix A. Concept 17.3 Eukaryotic cells modify RNA after transcription. Enzymes in the eukaryotic nucleus modify pre-mRNA in specific ways before the genetic message is dispatched to the cytoplasm. During this RNA processing, both ends of the primary transcript are altered. Also, in most cases, certain interior sections of the RNA molecule are cut out and the remaining parts spliced together. These modifications produce an mRNA molecule ready for translation. Alteration of mRNA ends. Each end of a pre-mRNA molecule is modified in a particular way. Figure 17.11. The 3 end, which is synthesized first, receives a 5 cap, a modified form of a guanine, G, nucleotide added onto the 5 end after transcription of the first 20 to 40 nucleotides have been transcribed. The 3 end of the pre-mRNA molecule is also modified before the mRNA exits the nucleus. Recall that the pre-mRNA is cut and released soon after the polyadenylation signal, AAUAAA, is transcribed. At the end, an enzyme then adds 50 to 250 more adenine, A, nucleotides, forming a poly-A tail. The cap and poly-A tail share several important functions. First, they seem to facilitate the export of the mature mRNA from the nucleus. Second, they help protect the mRNA from degradation by hydrolytic enzymes. And third, they help ribosomes attach to the end of the mRNA once it reaches the cytoplasm. In addition to the cap and tail on a eukaryotic mRNA molecule. Figure 17.11 shows the untranslated regions, UTRs, at the ends of the mRNA, referred to as the UTR and UTR. The UTRs are parts of the mRNA that will not be translated into protein, but they have other functions, such as ribosome binding. Figure 17.11 RNA processing, addition of the cap and poly-A tail. RNA, a single-stranded molecule, with 5' cap, a 5' UTR, region of protein coding segments, a 3' UTR with polyadenylation signal, and a poly-A tail. Enzymes modify the two ends of a eukaryotic pre-mRNA molecule. The modified ends may promote the export of mRNA from the nucleus, and they help protect the mRNA from degradation. When the mRNA reaches the cytoplasm, the modified ends, in conjunction with certain cytoplasmic proteins, help with ribosome attachment. The cap and poly-A tail are not translated into protein, nor are the regions called the untranslated region, UTR, and untranslated region, UTR. The pink segments are introns, which will be described shortly, see. Figure 17.12. Figure 17.11 Full alternative text. Mastering biology. Animation, adding the cap and poly-A tail. Split genes and RNA splicing. A remarkable stage of RNA processing in the eukaryotic nucleus is called RNA splicing. Figure 17.12, in which large portions of the RNA primary transcript molecules are removed and the remaining portions are reconnected. This cut and paste job is similar to editing a movie. The average length of a transcription unit along a human DNA molecule is about 27,000 nucleotide pairs, so the primary RNA transcript is also that long. However, the average sized protein of 400 amino acids requires only 1,200 nucleotides in RNA to code for it. Remember, each amino acid is encoded by a triplet of nucleotides. This is because most eukaryotic genes and their RNA transcripts have long non-coding stretches of nucleotides, regions that are not translated. Even more surprising is that most of these non-coding sequences are interspersed between coding segments of the gene and thus between coding segments of the pre-mRNA. In other words, the sequence of DNA nucleotides that codes for a eukaryotic polypeptide is usually not continuous, it is split into segments. The non-coding segments of nucleic acid that lie between coding regions are called intervening sequences, or introns. The other regions are called exons, because they are eventually expressed, usually by being translated into amino acid sequences. Exceptions include the UTRs of the exons at the ends of the RNA, which make up part of the mRNA but are not translated into protein. Because of these exceptions, you may prefer to think of exons as sequences of RNA that exit the nucleus. The terms intron and exon are used for both RNA sequences and the DNA sequences that specify them. Figure 17.12 RNA processing, RNA splicing. Pre-mRNA with 5' cap and 3' poly-A tail at the ends and segments of introns and exons in between. Arrow indicates introns are spliced out to form mRNA. The RNA molecule shown here codes for globin, one of the polypeptides of hemoglobin. The numbers under the RNA refer to codons. Globin is 146 amino acids long. The globin gene and its pre-mRNA transcript have 3 exons, corresponding to sequences that will exit the nucleus as mRNA. The UTR and UTR do not code for proteins, but they are parts of exons because they remain in the mRNA. During RNA processing, the introns are cut out and the exons spliced together. In many genes, the introns are much longer than the exons. Figure 17.12 Full alternative text. Draw IT on the mRNA, indicate the sites of the start and stop codons. Mastering biology. Bioflix registered animation, RNA processing. In making a primary transcript from a gene, RNA polymerase II transcribes both introns and exons from the DNA, but the mRNA molecule that enters the cytoplasm is an abridged version. In the process of RNA splicing, the introns are cut out from the molecule and the exons joined together, forming an mRNA molecule with a continuous coding sequence. How is pre-mRNA splicing carried out? The removal of introns is accomplished by a large complex made of proteins and small RNAs called a spliceosome. This complex binds to several short nucleotide sequences along an intron, including key sequences at each end. Figure 17.13. The intron is then released, and rapidly degraded, and the spliceosome joins together the 2 exons that flank the intron. The small RNAs in the spliceosome not only participate in spliceosome assembly and splice site recognition, but also catalyze the splicing reaction. Like proteins, RNAs can act as catalysts. Figure 17.13 A spliceosome splicing a pre-mRNA. Spliceosome drawn as a complex containing many small RNAs shown base pairing with regions on a long intron. On either side of the intron are exons. Intron is spliced out. The diagram shows a portion of a pre-mRNA transcript, with an intron, pink, flanked by 2 exons, red. Small RNAs within the spliceosome base pair with nucleotides at specific sites along the intron. Next, small spliceosome RNAs catalyze cutting of the pre-mRNA and the splicing together of the exons, releasing the intron for rapid degradation. Figure 17.13 Full alternative text. Mastering biology. Animation, a spliceosome. Mastering biology. Interview with Joan Steitz, studying RNA, her favorite molecule. Joan Steitz. Ribozymes. The idea of a catalytic role for the RNAs in the spliceosome arose from the discovery of ribozymes, RNA molecules that function as enzymes. In some organisms, RNA splicing can occur without proteins or even additional RNA molecules. The intron RNA functions as a ribozyme and catalyzes its own removal. For example, in the ciliate protist Tetrahymena, self-splicing occurs in the production of ribosomal RNA, rRNA, a component of the organism's ribosomes. The preRNA actually removes its own introns. The discovery of ribozymes invalidated the idea that all biological catalysts are proteins. 3 properties of RNA enable some RNA molecules to function as enzymes. First, because RNA is single-stranded, a region of an RNA molecule may base pair, in an anti-parallel arrangement, with a complementary region elsewhere in the same molecule. This gives the molecule a particular 3-dimensional structure. A specific structure is essential to the catalytic function of ribozymes, just as it is for enzymatic proteins. Second, like certain amino acids in an enzymatic protein, some of the bases in RNA contain functional groups that can participate in catalysis. Third, the ability of RNA to hydrogen bond with other nucleic acid molecules, either RNA or DNA, adds specificity to its catalytic activity. For example, complementary base pairing between the RNA of the spliceosome and the RNA of a primary RNA transcript precisely locates the region where the ribozyme catalyzes splicing. Later in this chapter, you will see how these properties of RNA also allow it to perform important non-catalytic roles in the cell, such as recognition of the 3 nucleotide codons on mRNA. The functional and evolutionary importance of introns. Evolution. Whether or not RNA splicing and the presence of introns have provided selective advantages during evolutionary history is a matter of some debate. In any case, it is informative to consider their possible adaptive benefits. Specific functions have not been identified for most introns, but at least some contain sequences that regulate gene expression, and many affect gene products. One important consequence of the presence of introns in genes is that a single gene can encode more than one kind of polypeptide. Many genes are known to give rise to 2 or more different polypeptides, depending on which segments are treated as exons during RNA processing. This is called alternative RNA splicing, see Figure 18.14. Results from the Human Genome Project, discussed in Concept 21.1, suggests that alternative RNA splicing is one reason humans can get along with about the same number of genes as a nematode, roundworm. Because of alternative splicing, the number of different protein products an organism produces can be much greater than its number of genes. Proteins often have a modular architecture consisting of discrete structural and functional regions called domains. One domain of an enzyme, for example, might include the active site, while another might allow the enzyme to bind to a cellular membrane. In quite a few cases, different exons code for the different domains of a protein. Figure 17.14. Figure 17.14 correspondence between exons and protein domains. Gene region on DNA with exon and intron regions labeled. Each exon region corresponds to a particular domain of the polypeptide that results from the expression of the gene. Figure 17.14 full alternative text. The presence of introns in a gene may facilitate the evolution of new and potentially beneficial proteins as a result of a process known as exon shuffling, see Figure 21.16. Introns increase the probability of crossing over between the exons of alleles of a gene simply by providing more terrain for crossovers without interrupting coding sequences. This might result in new combinations of exons and proteins with altered structure and function. We can also imagine the occasional mixing and matching of exons between completely different, non-allelic, genes. Exon shuffling of either sort could lead to new proteins with novel combinations of functions. While most of the shuffling would result in non-beneficial changes, occasionally a beneficial variant might arise. Concept check 17.3. Given that there are about 20,000 human protein coding genes, how can human cells make 75,000 to 100,000 different proteins? Compare RNA splicing to how you would watch a pre-recorded television show. What would introns correspond to in this analogy? What if? What would be the effect of treating cells with an agent that removed the cap from RNAs? For suggested answers, see Appendix A. Concept 17.4 translation is the RNA-directed synthesis of a polypeptide, a closer look. We will now examine how genetic information flows from mRNA to protein, the process of translation. Figure 17.15. We'll focus on the basic steps of translation that occur in both bacteria and eukaryotes, while pointing out key differences. Figure 17.15 Translation, the basic concept. mRNA inside ribosome. Transfer RNA binds to mRNA codon and transfers a PHE to growing polypeptide chain. Other tRNAs preparing to bind to mRNA codons and transfer their amino acids. As a molecule of mRNA is moved through a ribosome, codons are translated into amino acids, one by one. The translators, or interpreters, are tRNA molecules, each type with a specific anticodon at one end and a corresponding amino acid at the other end. A tRNA adds its amino acid cargo to a growing polypeptide chain when the anticodon hydrogen bonds to the complementary codon on the mRNA. Figure 17.15 Full alternative text. Mastering biology. Bioflix registered animation, translation. Animation, overview of translation. Molecular components of translation. In the process of translation, a cell reads a genetic message and builds a polypeptide accordingly. The message is a series of codons along an mRNA molecule, and the translator is called a transfer RNA, tRNA. The function of a tRNA is to transfer an amino acid from the cytoplasmic pool of amino acids to a growing polypeptide in a ribosome. A cell keeps its cytoplasm stocked with all 20 amino acids, either by synthesizing them from other compounds or by taking them up from the surrounding solution. The ribosome, a structure made of proteins and RNAs, adds each amino acid brought to it by a tRNA to the growing end of a polypeptide chain, see Figure 17.15. Translation is simple in principle but complex in its biochemistry and mechanics, especially in the eukaryotic cell. In dissecting translation, we'll focus on the slightly less complicated version of the process that occurs in bacteria. We'll first look at the major players in this process. The structure and function of transfer RNA. The key to translating a genetic message into a specific amino acid sequence is the fact that each tRNA molecule enables translation of a given mRNA codon into a certain amino acid. This is possible because a tRNA bears a specific amino acid at one end of its three-dimensional structure, while at the other end is a nucleotide triplet that can base pair with the complementary codon on mRNA. A tRNA molecule consists of a single RNA strand that is only about 80 nucleotides long, compared to hundreds of nucleotides for most mRNA molecules. The presence of complementary stretches of nucleotide bases that can hydrogen bond to each other allows the single strand to fold back on itself and form a molecule with a particular 3D structure. Flattened into one plane to clarify this base pairing, a tRNA molecule looks like a cloverleaf. Figure 17.16a. The tRNA actually twists and folds into a compact 3D structure that is roughly L-shaped. Figure 17.16b, with the ends of the linear tRNA both located near one end of the structure. The protruding end acts as the attachment site for an amino acid. The loop extending from the other end of the L includes the anticodon, the particular nucleotide triplet that base pairs to a specific mRNA codon. Thus, the structure of a tRNA molecule fits its function. Figure 17.16 The structure of transfer RNA, tRNA. The structure of transfer RNA, tRNA. Figure 17.16 Full alternative text. Visual skills. Look at the tRNA shown in this figure. Based on its anticodon, identify the codon it would bind to, as well as the amino acid that it would carry. Mastering biology. HHMI video, RNA folding HHMI. Anticodons are conventionally written to align properly with codons written, see Figure 17.15. For base pairing, RNA strands must be anti-parallel, like DNA. As an example of how tRNAs work, consider the mRNA codon, which is translated as the amino acid glycine. The tRNA that base pairs with this codon by hydrogen bonding has as its anticodon and carries glycine at its other end, see the incoming tRNA approaching the ribosome in Figure 17.15. As an mRNA molecule is moved through a ribosome, glycine will be added to the polypeptide chain whenever the codon is presented for translation. Codon by codon, the genetic message is translated as tRNAs position each amino acid in the order prescribed, and the ribosome adds that amino acid onto the growing polypeptide chain. The tRNA molecule is a translator in the sense that, in the context of the ribosome, it can read a nucleic acid word, the mRNA codon, and interpret it as a protein word, the amino acid. Like mRNA and other types of cellular RNA, transfer RNA molecules are transcribed from DNA templates. In a eukaryotic cell, tRNA, like mRNA, is made in the nucleus and then travels to the cytoplasm, where it will participate in the process of translation. In both bacterial and eukaryotic cells, each tRNA molecule is used repeatedly, picking up its designated amino acid in the cytosol, depositing this cargo onto a polypeptide chain at the ribosome, and then leaving the ribosome, ready to pick up another of the same amino acid. The accurate translation of a genetic message requires two instances of molecular recognition. First, a tRNA that binds to an mRNA codon specifying a particular amino acid must carry that amino acid, and no other, to the ribosome. The correct matching up of tRNA and amino acid is carried out by a family of related enzymes that are aptly named aminoacylgranous synthetases. Figure 17.17. The active site of each type of aminoacylgranous synthetase fits only a specific combination of amino acid and tRNA. Regions of both the amino acid attachment end and the anticodon end of the tRNA ensure the specific fit. There are 20 different synthetases, one for each amino acid. A synthetase joins a given amino acid to an appropriate tRNA. One synthetase is able to bind to all the different tRNAs for its particular amino acid. The synthetase catalyzes the covalent attachment of the amino acid to its tRNA in a process driven by the hydrolysis of ADP. The resulting aminoacyl tRNA, also called a charged tRNA, is released from the enzyme and is then available to deliver its amino acid to a growing polypeptide chain on a ribosome. Figure 17.17. Aminoacylgranous synthetases provide specificity in joining amino acids to their tRNAs. Aminoacyl tRNA synthase links tRNA and its specific amino acid with covalent bonding using ATP. Linkage of a tRNA to its amino acid is an endergonic process that occurs at the expense of ADP, which loses two phosphate groups, becoming AMP. Figure 17.17 Full alternative text. Mastering biology. Animation, transfer RNA. The second instance of molecular recognition is the pairing of the tRNA anticodon with the appropriate mRNA codon. If one tRNA type existed for each mRNA codon specifying an amino acid, there would be 61 tRNAs, see Figure 17.6. In bacteria, however, there are only about 45 tRNAs, signifying that some tRNAs must be able to bind to more than one codon. This is possible because the rules for base pairing between the third nucleotide base of a codon and the corresponding base of a tRNA anticodon are relaxed compared to those at other codon positions. For example, the nucleotide base U at the end of a tRNA anticodon can pair with either A or G in the third position, at the end, of an mRNA codon. The flexible base pairing at this codon position is called wobble. Wobble explains why the synonymous codons for a given amino acid most often differ in their third nucleotide base. Accordingly, a tRNA with the anticodon can base pair with either the mRNA codon or, both of which code for arginine, see Figure 17.6. The structure and function of ribosomes. Although the ribosomes of bacteria and eukaryotes are very similar in structure and function, eukaryotic ribosomes are slightly larger and differ somewhat from bacterial ribosomes in their molecular composition. The differences are medically significant. Certain antibiotic drugs can inactivate bacterial ribosomes without affecting the ability of eukaryotic ribosomes to make proteins. These drugs, including tetracycline and streptomycin, are used to combat bacterial infections. Ribosomes facilitate the specific coupling of tRNA anticodons with mRNA codons during protein synthesis. A ribosome consists of a large subunit and a small subunit, each made up of proteins and one or more ribosomal RNAs, or rRNAs. Figure 17.18. In eukaryotes, the subunits are made in the nucleolus. Ribosomal RNA genes are transcribed, and the RNA is processed and assembled with proteins imported from the cytoplasm. Completed ribosomal subunits are then exported via nuclear pores to the cytoplasm. In both bacteria and eukaryotes, a large and a small subunit join to form a functional ribosome only when attached to an mRNA molecule. About 1/3 of the mass of a ribosome is made up of proteins, the rest consists of 3 rRNA molecules, in bacteria, or 4, in eukaryotes. Because most cells contain thousands of ribosomes, rRNA is the most abundant type of cellular RNA. Figure 17.18 The anatomy of a functioning ribosome. The anatomy of a functioning ribosome. Figure 17.18 Full alternative text. Mastering biology. Animation, ribosomes. The structure of a ribosome reflects its function of bringing an mRNA molecule together with tRNAs carrying amino acids. The mRNA itself has a binding site for the ribosome. In the scientific skills exercise, you can work with DNA sequences representing this binding site in a group of Escherichia coli genes. The ribosome, in turn, has a binding site for mRNA, as well as 3 binding sites for tRNA, see Figure 17.18. The P site, peptidyltRNA binding site, holds the tRNA carrying the growing polypeptide chain, while the A site, amino acyltRNA binding site, holds the tRNA carrying the next amino acid to be added to the chain. Discharged tRNAs leave the ribosome from the E site, exit site. The ribosome holds the tRNA and mRNA in close proximity and positions the new amino acid so that it can be added to the carboxyl end of the growing polypeptide. It then catalyzes the formation of the peptide bond. As the polypeptide becomes longer, it passes through an exit tunnel in the ribosome's large subunit. When the polypeptide is complete, it is released through the exit tunnel. The widely accepted model is that rRNAs, rather than ribosomal proteins, are primarily responsible for both the structure and the function of the ribosome. The proteins, which are largely on the exterior, support the shape changes of the rRNA molecules as they carry out catalysis during translation. Ribosomal RNA is the main constituent of the A and P sites and of the interface between the two subunits. It also acts as the catalyst of peptide bond formation. Thus, a ribosome could actually be considered one colossal ribozyme. Building a polypeptide. We can divide translation, the synthesis of a polypeptide, into 3 stages: initiation, elongation, and termination. All 3 require protein factors that aid in the translation process. Some steps of initiation and elongation also require energy, provided by the hydrolysis of guanosine triphosphate, GTP. Ribosome association and initiation of translation. The initiation stage of translation brings together an mRNA, a tRNA bearing the first amino acid of the polypeptide, and the two subunits of a ribosome. First, a small ribosomal subunit binds to both the mRNA and a specific initiator tRNA, which carries the amino acid methionine. In bacteria, the small subunit can bind the two in either order. It binds the mRNA at a specific RNA sequence, just upstream of the OX-start codon. In eukaryotes, the small subunit, with the initiator tRNA already bound, binds to the cap of the mRNA and then moves, or scans, downstream along the mRNA until it reaches the OX-start codon, where the initiator tRNA then hydrogen bonds, as shown in step 1 of Figure 17.19. In either bacteria or eukaryotes, the OX signals the start of translation. This is important because it establishes the codon reading frame for the mRNA. Figure 17.19. The initiation of translation. The initiation of translation. Figure 17.19. Full alternative text. Mastering biology. Animation, initiation of translation. Scientific skills exercise. Interpreting a sequence logo. How can a sequence logo be used to identify ribosome binding sites on bacterial RNAs? When initiating translation, ribosomes bind to an mRNA at a ribosome binding site upstream of the OX-start codon. Because RNAs from different genes all bind to a ribosome, the genes encoding these RNAs are likely to have a similar base sequence where the ribosomes bind. Therefore, candidate ribosome binding sites on mRNA can be identified by comparing DNA sequences, and thus the mRNA sequences, of multiple genes in a species, searching the region upstream of the start codon for shared, conserved, stretches of bases. In this exercise, you will analyze DNA sequences from multiple such genes, represented by a visual graphic called a sequence logo. The ribosome binding site on mRNA is toward the 5' end. How the experiment was done. The DNA sequences of 149 genes from the E. coli genome were aligned using computer software. The aim was to identify similar base sequences at the appropriate location in each gene as potential ribosome binding sites. Rather than presenting the data as a series of 149 sequences aligned in a column, a sequence alignment, the researchers used a sequence logo. Data from the experiment to show how sequence logos are made. The potential ribosome binding regions from 10 E. coli genes are shown below in a sequence alignment, followed by the sequence logo derived from the aligned sequences. Note that the DNA shown is the non-template, coding, strand, which is how DNA sequences are typically presented. All data from Thomas D. Schneider. Sequence logo derived from the aligned sequences of 10 E. coli genes. Sequence alignment. 17.4 to 10. Full alternative text. Letters are placed on a horizontal axis labeled 5' to 3' from left to right. The tallest part of the sequence is AGT at 0, T at 1, and G at 2. Sequence logo. Interpret the data. In the sequence logo, the horizontal axis shows the primary sequence of the DNA by nucleotide position. Letters for each base are stacked on top of each other according to their relative frequency at that position among the aligned sequences, with the most common base as the largest letter at the top of the stack. The height of each letter represents the relative frequency of that base at that position. A. In the sequence alignment, count the number of each base at position 9 and order them from most to least frequent. Compare this to the size and placement of each base at 9 in the logo. B. Do the same for positions 0 and 1. The height of a stack of letters in a logo indicates the predictive power of that stack, determined statistically. If the stack is tall, we can be more confident in predicting what base will be in that position if a new sequence is added to the logo. For example, at position 2 in the sequence alignment, all 10 sequences have AG. The probability of finding AG there in a new sequence is very high, as is the stack in the sequence logo. For short stacks, the bases all have about the same frequency, so it's hard to predict a base at those positions. A. Looking at the sequence logo, which two positions have the most predictable bases? What bases do you predict would be at those positions in a newly sequenced gene? B. Which 12 positions have the least predictable bases? How do you know? How does this reflect the relative frequencies of the bases shown at these positions in the sequence alignment? Use the two leftmost positions of the 12 as examples in your answer. In the actual experiment, the researchers used 149 sequences to build their sequence logo, which is shown below. There is a stack at each position, even if short, because the sequence logo includes more data. A. Which 3 positions in the sequence logo have the most predictable bases? Name the most frequent base at each. B. Which 4 positions have the least predictable bases? How can you tell? Letters are placed on a horizontal axis labeled 5' to 3' from left to right. The tallest part of the sequence is AGT at 0, T at 1, and G at 2. Further reading T.D. Schneider and R.M. Stevens, Sequence Logos, A New Way to Display Consensus Sequences, Nucleic Acids Research 18:6097, 6100, 1990. A consensus sequence identifies the base occurring most often at each position in the set of sequences. A. Write out the consensus sequence of this, the non-template, strand. In any position where the base can't be determined, put a dash. B. Which provides more information, the consensus sequence or the sequence logo. What is lost in the less informative method? A. Based on the logo, what 5 adjacent base positions in the UTR region are most likely to be involved in ribosome binding? Explain. B. What is represented by the bases in positions 0 to 2? Instructors, a version of the scientific skills exercise can be assigned in Mastering Biology. The union of mRNA, initiator tRNA, and the small ribosomal subunit is followed by the attachment of a large ribosomal subunit, completing the translation initiation complex, C. C. Figure 17.19. Proteins called initiation factors are required to bring all these components together. The cell also expends energy obtained by hydrolysis of a GTP molecule to form the initiation complex. At the completion of the initiation process, the initiator tRNA sits in the P site of the ribosome, and the vacant A site is ready for the next amino acyl tRNA. Note that a polypeptide is always synthesized in one direction, from the initial methionine at the amino end, also called the N-terminus, toward the final amino acid at the carboxyl end, also called the C-terminus, C. Figure 5.15. Elongation of the polypeptide chain. In the elongation stage of translation, amino acids are added one by one to the previous amino acid at the C-terminus of the growing chain. Each addition involves several proteins called elongation factors and occurs in a three-step cycle described in Figure 17.20. Energy expenditure occurs in the first and third steps. Codon recognition requires hydrolysis of one molecule of GTP, which increases the accuracy and efficiency of this step. One more GTP is hydrolyzed, broken up, to provide energy for the translocation step. Figure 17.20. The elongation cycle of translation. The elongation cycle of translation. The hydrolysis of GTP plays an important role in the elongation process. Elongation factors are not shown. Figure 17.20. Full alternative text. Mastering biology. Animation, elongation cycle of translation. The mRNA is moved through the ribosome in one direction only, and first, this is equivalent to the ribosome moving on the mRNA. The main point is that the ribosome and the mRNA move relative to each other, unidirectionally, codon by codon. The elongation cycle takes less than 1/10 of a second in bacteria and is repeated as each amino acid is added until the polypeptide is completed. The empty tRNAs that are released from the E site return to the cytoplasm, where they will be reloaded with the appropriate amino acid, see Figure 17.17. Termination of translation. The final stage of translation is termination. Figure 17.21. Elongation continues until a stop codon in the mRNA reaches the A site. The nucleotide-based triplets UAG, UAA, and UGA, all written, do not code for amino acids but instead act as signals to stop translation. A release factor, a protein shaped like an amino acyl tRNA, binds directly to the stop codon in the A site. The release factor causes the addition of a water molecule instead of an amino acid to the polypeptide chain. Water molecules are abundant in the cytosol. This reaction hydrolyzes, breaks, the bond between the completed polypeptide and the tRNA in the P site, releasing the polypeptide through the exit tunnel of the ribosome's large subunit. The remainder of the translation assembly then comes apart in a multi-step process, aided by other protein factors. Breakdown of the translation assembly requires the hydrolysis of 2 more GTP molecules. Figure 17.21. The termination of translation. The termination of translation. Like elongation, termination requires GTP hydrolysis as well as additional protein factors, which are not shown here. Figure 17.21. Full alternative text. Mastering biology. Animation, termination of translation. Completing and targeting the functional protein. The process of translation is often not sufficient to make a functional protein. In this section, you will learn about modifications that polypeptide chains undergo after the translation process as well as some of the mechanisms used to target completed proteins to specific sites in the cell. Protein folding and post-translational modifications. During its synthesis, a polypeptide chain begins to coil and fold spontaneously as a consequence of its amino acid sequence, primary structure, forming a protein with a specific shape, a three-dimensional molecule with secondary and tertiary structure, see Figure 5.18. Thus, a gene determines primary structure, which in turn determines shape. Additional steps post-translational modifications may be required before the protein can begin doing its particular job in the cell. Certain amino acids may be chemically modified by the attachment of sugars, lipids, phosphate groups, or other additions. Enzymes may remove one or more amino acids from the leading, amino, end of the polypeptide chain. In some cases, a polypeptide chain may be enzymatically cleaved into 2 or more pieces. In other cases, 2 or more polypeptides that are synthesized separately may come together, if the protein has quaternary structure. An example is hemoglobin, see Figure 5.18. Mastering biology. Bioflix registered animation, protein processing. Targeting polypeptides to specific locations. In electron micrographs of eukaryotic cells active in protein synthesis, 2 populations of ribosomes are evident, free and bound, see Figure 6.10. Free ribosomes are suspended in the cytosol and mostly synthesize proteins that stay in the cytosol and function there. In contrast, bound ribosomes are attached to the cytosolic side of the endoplasmic reticulum, ER, or to the nuclear envelope. Bound ribosomes make proteins of the endomembrane system, see Figure 6.15, as well as proteins secreted from the cell, such as insulin. It is important to note that the ribosomes themselves are identical and can alternate between being free ribosomes one time they are used and being bound ribosomes the next. What determines whether a ribosome is free in the cytosol or bound to rough ER? Polypeptide synthesis always begins in the cytosol as a free ribosome starts to translate an mRNA molecule. There, the process continues to completion unless the growing polypeptide itself cues the ribosome to attach to the ER. The polypeptides of proteins destined for the endomembrane system or for secretion are marked by a signal peptide, which targets the protein to the ER. Figure 17.22. The signal peptide, a sequence of about 20 amino acids at or near the leading end, N-terminus, of the polypeptide, is recognized as it emerges from the ribosome by a protein RNA complex called a signal recognition particle, SRP. This particle escorts the ribosome to a receptor protein built into the ER membrane. The receptor is part of a multi-protein translocation complex. Polypeptide synthesis continues there, and the growing polypeptide snakes across the membrane into the ER lumen via a protein pore. The signal peptide is removed by an enzyme. The rest of the completed polypeptide, if it is to be secreted from the cell, is released into solution within the ER lumen, see Figure 17.22. Or, if the polypeptide is to be a membrane protein, amino acid sequences further in the chain cause that part to remain embedded in the ER membrane. In either case, it travels in a transport vesicle to its destination, see Figure 7.9. Figure 17.22. The signal mechanism for targeting proteins to the ER. The signal mechanism for targeting proteins to the ER. Figure 17.22. Full alternative text. Make connections. If this protein were destined for secretion, what would happen to it after its synthesis was completed? See Figure 7.9. Mastering biology. Animation, co-translational translocation. Other kinds of signal peptides are used to target polypeptides to mitochondria, chloroplasts, the interior of the nucleus, and other organelles that are not part of the endomembrane system. The critical difference in these cases is that translation is completed in the cytosol before the polypeptide is imported into the organelle. Translocation mechanisms also vary, but in all cases studied to date, the postal zip codes that address proteins for secretion or to cellular locations are signal peptides of some sort. Bacteria also employ signal peptides to target proteins to the plasma membrane or for secretion. Making multiple polypeptides in bacteria and eukaryotes. In previous sections, you learned how a single polypeptide is synthesized using the information encoded in an mRNA molecule. When a polypeptide is required in a cell, though, the need is for many copies, not just one. In both bacteria and eukaryotes, multiple ribosomes translate an mRNA at the same time. Figure 17.23, that is, a single mRNA is used to make many copies of a polypeptide simultaneously. Once a ribosome is far enough past the start codon, a second ribosome can attach to the mRNA, eventually resulting in a number of ribosomes trailing along the mRNA. Such strings of ribosomes, called polyribosomes, or polysomes, can be seen with an electron microscope. They can be either free or bound. They enable a cell to rapidly make many copies of a polypeptide. Figure 17.23. Polyribosomes. Polyribosomes are diagrammed. Figure 17.23. Full alternative text. Another way both bacteria and eukaryotes increase the number of copies of a polypeptide is by transcribing multiple RNAs from the same gene. However, the coordination of the two processes transcription and translation differs in the two groups. The most important differences between bacteria and eukaryotes arise from the bacterial cell's lack of compartmental organization. Like a one-room workshop, a bacterial cell ensures a streamlined operation by coupling the two processes. With no nuclear envelope, it can simultaneously transcribe and translate the same gene. Figure 17.24, and the newly made protein can quickly diffuse to its site of function. Figure 17.24 Coupled transcription and translation in bacteria. Coupled transcription and translation in bacteria. Micrograph, TEM, of strand of coupled transcription and translation in E. coli DNA, scale, 0.25 micrometers, with diagram detailing this process. In bacterial cells, the translation of mRNA can begin as soon as the leading end of the mRNA molecule peels away from the DNA template. The micrograph, TEM, shows a strand of E. coli DNA being transcribed by RNA polymerase molecules. Attached to each RNA polymerase molecule is a growing strand of mRNA, which is already being translated by ribosomes. The newly synthesized polypeptides are not visible in the micrograph but are shown in the diagram. Figure 17.24 Full alternative text. Visual skills. Which one of the mRNA molecules started being transcribed first? On that mRNA, which ribosome started translating the mRNA first? In contrast, the eukaryotic cell's nuclear envelope segregates transcription from translation and provides a compartment for extensive RNA processing. This processing stage includes additional steps, discussed earlier, the regulation of which can help coordinate the eukaryotic cell's elaborate activities. Figure 17.25 summarizes the path from gene to polypeptide in a eukaryotic cell. Figure 17.25 A summary of transcription and translation in a eukaryotic cell. A summary of transcription and translation in a eukaryotic cell. This diagram shows the path from one gene to one polypeptide. Each gene in the DNA can be transcribed repeatedly into many identical RNA molecules, and each mRNA can be translated repeatedly to yield many identical polypeptide molecules. Remember that the final products of some genes are not polypeptides but RNA molecules that don't get translated, including tRNAs and rRNAs. In general, the steps of transcription and translation are similar in bacterial, archaeal, and eukaryotic cells. The major difference is the occurrence of RNA processing in the eukaryotic nucleus. Other significant differences are found in the initiation stages of both transcription and translation and in the termination of transcription. To visualize these processes in their cellular context, see Figure 6.32d. F. Figure 17.25 Full alternative text. Mastering biology. Bioflix registered animation, protein synthesis. Concept check 17.4. What two processes ensure that the correct amino acid is added to a growing polypeptide chain? Describe how a polypeptide to be secreted reaches the endomembrane system. Draw a tRNA with the anticodon. Given wobble, what two different codons could it bind to? Draw each codon on an mRNA, labeling all N-ends, the tRNA, and the amino acid it carries. What if? In eukaryotic cells, mRNAs have been found to have a circular arrangement in which proteins hold the poly-A tail near the cap. How might this increase translation efficiency? For suggested answers, see Appendix A. Concept 17.5 Mutations of one or a few nucleotides can affect protein structure and function. Now that you have explored the process of gene expression, you can consider the effects of changes to the genetic information of a cell. Because these changes, called mutations, are the ultimate source of new genes, they are responsible for the huge diversity of genes found among organisms. Earlier, we considered chromosomal rearrangements that affect long segments of DNA, see Figure 15.14, these are considered large-scale mutations. Here we examine small-scale mutations of one or a few nucleotide pairs, including point mutations, changes in a single nucleotide pair of a gene. If a point mutation occurs in a gamete or in a cell that gives rise to gametes, it may be transmitted to offspring and to future generations. If the mutation has an adverse effect on the phenotype of a person, the mutant condition is referred to as a genetic disorder or hereditary disease. For example, we can trace the genetic basis of sickle cell disease to the mutation of a single nucleotide pair in the gene that encodes the beta-globin polypeptide of hemoglobin. The change of a single nucleotide in the DNA's template strand leads to an altered mRNA and the production of an abnormal protein. Figure 17.26, also see Figure 5.19. In individuals who are homozygous for the mutant allele, the sickling of red blood cells caused by the altered hemoglobin produces the multiple symptoms associated with sickle cell disease, see Concept 14.4 End. Figure 23.18. Another disorder caused by a point mutation is a heart condition called familial cardiomyopathy, which is responsible for some of the tragic incidents of sudden death in young athletes. Point mutations in several genes encoding muscle proteins have been identified, any of which can lead to this disorder. Figure 17.26 The molecular basis of sickle cell disease, a point mutation. The molecular basis of sickle cell disease. The allele that causes sickle cell disease differs from the wild-type, normal, allele by a single DNA nucleotide pair. The micrographs are stems of a normal red blood cell, on the left, and a sickled red blood cell, right, from individuals homozygous for wild-type and mutant alleles, respectively. Figure 17.26 Full alternative text. Mastering Biology. HHMI Animation, Sickle Cell Disease HHMI. Types of small-scale mutations. Many mutations occur in regions outside of protein-coding genes, and any potential effect they have on the phenotype of the organism may be subtle and hard to detect. For this reason, here we'll concentrate on mutations within protein-coding genes. Small-scale mutations within a gene can be divided into two general categories: 1. Single nucleotide pair substitutions and 2. Nucleotide pair insertions or deletions. Insertions and deletions can involve one or more nucleotide pairs. Substitutions. A. Nucleotide pair substitution is the replacement of one nucleotide and its partner with another pair of nucleotides. Figure 17.27a. Some substitutions have no effect on the encoded protein, owing to the redundancy of the genetic code. For example, if 3CCG5 on the template strand mutated to 3CCA5, the mRNA codon that used to be GGC would become GGU, but a glycine would still be inserted at the proper location in the protein, see Figure 17.6. In other words, a change in a nucleotide pair may transform one codon into another that is translated into the same amino acid. Such a change is an example of a silent mutation, which has no observable effect on the phenotype. Silent mutations can occur outside genes as well. Interestingly, there is evidence that some silent mutations may indirectly affect where or at what level the gene gets expressed, even though the actual protein is the same. Figure 17.27 Types of small-scale mutations that affect mRNA sequence. Wild type. Transcription of DNA template strand leads to normal mRNA and a normal protein, here, MET, LIS, PHE, GLY. All but one of the types shown here also affect the amino acid sequence of the encoded polypeptide. Figure 17.27 Full alternative text. Mastering Biology. Figure walkthrough animation, mutation types. Substitutions that change one amino acid to another one are called missense mutations. Such a mutation may have little effect on the protein, the new amino acid may have properties similar to those of the amino acid it replaces, or it may be in a region of the protein where the exact sequence of amino acids is not essential to the protein's function. However, the nucleotide pair substitutions of greatest interest are those that cause a major change in a protein. The alteration of a single amino acid in a crucial area of a protein, such as in the part of the beta-globin subunit of hemoglobin shown in Figure 17.26 or in the active site of an enzyme as shown in Figure 8.19 can significantly alter protein activity. Occasionally, such a mutation leads to an improved protein or one with novel capabilities, but much more often such mutations are neutral or detrimental, leading to a useless or less active protein that impairs cellular function. A second example of a missense mutation is a mutation in the tyrosinase gene that causes the albino phenotype in the Asinara donkey. Recently, Italian researchers sequenced the tyrosinase gene in donkeys from this wild population and showed that a recessive mutation is responsible for the lack of pigment. AC is changed to AG in this gene, resulting in a histidine being added to the protein instead of an aspartic acid. The changed amino acid is in a site in tyrosinase that normally binds a copper atom, and it makes the enzyme incapable of binding copper, rendering it non-functional. As in the case of sickle cell disease, just one change in a nucleotide base results in a dramatic phenotypic change. Substitution mutations are usually missense mutations, that is, the altered codon still codes for an amino acid and thus makes sense, although not necessarily the right sense. But a point mutation can also change a codon for an amino acid into a stop codon. This is called a nonsense mutation, and it causes translation to be terminated prematurely. The resulting polypeptide will be shorter than the polypeptide encoded by the normal gene. Most nonsense mutations lead to non-functional proteins. In the problem-solving exercise, you'll work with a few common single nucleotide pair substitution mutations in the gene encoding insulin, some or all of which may lead to diabetes. You will classify these mutations into one of the types we just described and characterize the change in the amino acid sequence. Insertions and deletions. Insertions and deletions are additions or losses of nucleotide pairs in a gene. Figure 17.27b. These mutations have a disastrous effect on the resulting protein more often than substitutions do. Insertion or deletion of nucleotides may alter the reading frame of the genetic message, the triplet grouping of nucleotides on the mRNA that is read during translation. Such a mutation, called a frameshift mutation, occurs whenever the number of nucleotides inserted or deleted is not a multiple of 3. All nucleotides downstream of the deletion or insertion will be improperly grouped into codons. The result will be extensive missense mutations, usually ending sooner or later in a nonsense mutation that leads to premature termination. Unless the frameshift is very near the end of the gene, the protein is almost certain to be non-functional. Insertions and deletions also occur outside of coding regions. These are not called frameshift mutations, but can have effects on the phenotype. For instance, they can affect how a gene is expressed. Problem-solving exercise. Are insulin mutations the cause of 3 infants' neonatal diabetes? Are insulin mutations the cause of 3 infants' neonatal diabetes? Insulin is a hormone that acts as a key regulator of blood glucose level, and insulin deficiency can lead to diabetes. In some cases of neonatal, newborn, diabetes, the gene coding for the insulin protein has a nucleotide pair substitution mutation that alters the protein structure enough to cause it to malfunction. Doctors can use DNA sequence information to diagnose diabetes and other diseases. For example, the insulin gene sequence of a patient with neonatal diabetes can be analyzed to determine if it has a mutation and, if so, its effect. In this exercise, you will determine the effect of mutations within insulin gene sequences in infant diabetes patients. Your approach: suppose you are a medical geneticist presented with 3 infant patients, all of whom have a nucleotide pair substitution in their insulin gene. It is your job to analyze each mutation to figure out its effect on the amino acid sequence of the insulin protein. To identify the mutation in each patient, you will compare his or her individual insulin complementary DNA, cDNA, sequence to that of the wild-type cDNA. cDNA is a double-stranded DNA molecule that is based on the mRNA sequence and thus contains only the portion of a gene that is translated. Introns are not included. cDNA sequences are commonly used to compare the coding regions of genes. Identifying the codons that have been changed will tell you which, if any, amino acids are altered in the patient's insulin protein. Your data: you will analyze the cDNA codons for amino acids 35 to 54, of the 110 amino acids, of each patient's insulin protein, so the start codon, OG, is not present. The sequences of the wild-type cDNA and the patient's cDNA are shown below, arranged in codons. The sequences of the wild-type cDNA and the patient's cDNA are shown below, arranged in codons. Data from N. Nishi and K. Nanjo, Insulin Gene Mutations and Diabetes, Journal of Diabetes Investigation 2:92, 100, 2011. 17.5 to 14 Full alternative text. Your analysis. Compare each patient's cDNA sequence to the wild-type cDNA sequence. Circle the codons where a nucleotide pair substitution mutation has occurred. Use a codon table, see Figure 17.6, to identify the amino acid encoded by the codon with the mutation in each patient's insulin sequence, and compare it to the amino acid encoded by the codon in the corresponding wild-type sequence. As is standard practice with DNA sequences, the coding, non-template, strand of the cDNA has been provided, so to convert it to mRNA for use with the codon table, you only need to change T to U. Classify each patient's nucleotide pair substitution mutation, is it a silent, missense, or nonsense mutation? Explain, for each answer. Compare the structure of the amino acid you identified in each patient's insulin sequence to that of the corresponding amino acid in the wild-type sequence, see Figure 5.14. Given that each patient has neonatal diabetes, discuss how the change of amino acid in each might have affected the insulin protein and thus resulted in the disease. Consider the chemical nature of the side chains. Instructors, a version of this problem-solving exercise can be assigned in Mastering Biology. Or a more extensive investigation called Solve It, which insulin mutations may result in disease, can be assigned. New mutations and mutagens. Mutations can arise in a number of ways. Errors during DNA replication or recombination can lead to nucleotide pair substitutions, insertions, or deletions, as well as to mutations affecting longer stretches of DNA. If an incorrect nucleotide is added to a growing chain during replication, for example, the base on that nucleotide will then be mismatched with the nucleotide base on the other strand. In many cases, the error will be corrected by DNA proofreading and repair systems, see Concept 16.2. Otherwise, the incorrect base will be used as a template in the next round of replication, resulting in a mutation. Such mutations are called spontaneous mutations. It is difficult to calculate the rate at which such mutations occur. Rough estimates have been made of the rate of mutation during DNA replication for both E. coli and eukaryotes, and the numbers are similar: about 1 nucleotide in every 1010 is altered, and the change is passed on to the next generation of cells. A number of physical and chemical agents, called mutagens, interact with DNA in ways that cause mutations. In the 1920s, Hermann Muller discovered that X-rays cause genetic changes in fruit flies, and he used X-rays to make Drosophila mutants for his genetic studies, just as Beadle and Tatum did with their neurospora cells. But Muller also recognized an alarming implication of his discovery: X-rays and other forms of high-energy radiation pose hazards to the genetic material of people as well as laboratory organisms. Mutagenic radiation, a physical mutagen, includes ultraviolet, UV, light, which can cause disruptive thymine dimers in DNA, see Figure 16.20. Chemical mutagens fall into several categories. Nucleotide analogues are chemicals similar to normal DNA nucleotides but that pair incorrectly during DNA replication. Other chemical mutagens interfere with correct DNA replication by inserting themselves into the DNA and distorting the double helix. Still other mutagens cause chemical changes in bases that change their pairing properties. Researchers have developed a variety of methods to test the mutagenic activity of chemicals. A major application of these tests is the preliminary screening of chemicals to identify those that may cause cancer. This approach makes sense because most carcinogens, cancer-causing chemicals, are mutagenic, and conversely, most mutagens are carcinogenic. Using CRISPR to edit genes and correct disease-causing mutations. Ever since biologists have understood how disease-causing proteins were the result of mutations in genes, they have sought techniques for gene editing, altering genes in a specific, predictable way. Their aim has been to use such a technique to change specific genes in living cells, in part to try to correct genes that cause disease. Over the past 15 years, biologists have developed a powerful new technique for gene editing, called the CRISPR-Cas9 system, that is transforming the field of genetic engineering, the direct manipulation of genes for practical purposes. Cas9 is a bacterial protein that helps defend bacteria against the viruses that infect them, bacteriophages. In bacterial cells, Cas9 acts together with guide RNA made from the CRISPR region of the bacterial genome. Figure 19.7 explains how this defense system works in bacteria. Mastering Biology. HHMI video, Jennifer Doudna, CRISPR provides a tool for changing genes. HHMI. Mastering Biology. Interview with Francisco Mojica, Discovering CRISPR in Bacteria, see the interview before. Chapter 13. Francisco Mojica. similar to other DNA cutting enzymes involved in DNA repair, described in Concept 16.2, Ca9 is a nuclease that cuts double-stranded DNA molecules. The power of Ca9 for gene editing is that the Ca9 protein will cut any sequence to which it is directed. Ca9 is directed to its target by a guide RNA molecule that it binds and uses as a homing device, cutting both strands of any DNA sequence that is complementary to the guide RNA. Scientists have been able to exploit the function of Ca9 by introducing a Ca9 guide RNA complex into a cell they wish to alter. Figure 17.28. The guide RNA in the complex is engineered to be complementary to the target gene. Ca9 cuts both strands of the target DNA, and the resulting broken ends of DNA trigger a DNA repair system, similar to that shown in Figure 16.20. When there is no undamaged DNA for the enzymes of the repair system to use as a template, as shown at the bottom left of Figure 17.28, the repair enzymes introduce or remove random nucleotides while rejoining the ends. Generally, this process alters the DNA sequence so that the gene no longer works properly. This technique is a highly successful way for researchers to knock out, disable, a given gene to study what that gene does in an organism. Figure 17.28 Gene editing using the CRISPR-Cas9 system. Gene editing using the CRISPR-Cas9 system. Figure 17.28 Full alternative text. But what about using this system to help treat genetic diseases? Researchers have modified the technique so that the CRISPR-Cas9 system can be used to repair a gene that has a harmful mutation. They introduce a segment from the normal, functional, gene along with the CRISPR-Cas9 system. After Ca9 cuts the target DNA, repair enzymes can use the normal DNA as a template to repair the target DNA at the breakpoint. In this way, the CRISPR-Cas9 system edits the defective gene so that it is corrected. See the bottom right of Figure 17.28. In 2018, researchers reported promising results using the CRISPR-Cas9 system in an attempt to correct the genetic defect that causes sickle cell disease. They edited human cells from patients with sickle cell disease and injected them into bone marrow in mice. After 19 weeks, the gene remained corrected in 20 to 40% of the injected cells. Researchers, physicians, and patients alike are excited about the potential of CRISPR technology to treat or even cure human diseases that have a genetic basis, such as sickle cell, Alzheimer's, and Parkinson's diseases, as well as some types of cancer. However, there are still concerns about using CRISPR-treated cells in humans because of possible effects on genes that are not being targeted. This is an active area of research. Jennifer Doudna, a co-discoverer of CRISPR-Cas9, recognized not only its incredible potential but also the danger of its misapplication. After having a dream in which Adolf Hitler asked her what could be done with this gene editing tool, Doudna realized it was imperative to spend some time reflecting on ethical considerations. In 2015, she convened a conference of biologists who agreed at the end of the meeting to urge extreme caution as the field moves forward and the discussion continues. What is a gene? Revisiting the question. Our definition of a gene has evolved over the past few chapters, as it has through the history of genetics. We began with the Mendelian concept of a gene as a discrete unit of inheritance that affects a phenotypic character. Chapter 14. We saw that Morgan and his colleagues assigned such genes to specific loci on chromosomes. Chapter 15. We went on to view a gene as a region of specific nucleotide sequence along the length of the DNA molecule of a chromosome. Chapter 16. Finally, in this chapter, we have considered a functional definition of a gene as a DNA sequence that codes for a specific polypeptide chain or a functional RNA molecule, such as a tRNA. All these definitions are useful, depending on the context in which genes are being studied. We have noted that merely saying a gene codes for a polypeptide is an oversimplification. Most eukaryotic genes contain non-coding segments, such as introns, so large portions of these genes have no corresponding segments in polypeptides. Molecular biologists also often include promoters and certain other regulatory regions of DNA within the boundaries of a gene. These DNA sequences are not transcribed, but they can be considered part of the functional gene because they must be present for transcription to occur. Our definition of a gene must also be broad enough to include the DNA that is transcribed into rRNA, tRNA, and other RNAs that are not translated. These genes have no polypeptide products but play crucial roles in the cell. Thus, we arrive at the following definition: a gene is a region of DNA that can be expressed to produce a final functional product that is either a polypeptide or an RNA molecule. When considering phenotypes, however, it is often useful to start by focusing on genes that code for polypeptides. In this chapter, you have learned in molecular terms how a typical gene is expressed by transcription into RNA and then translation into a polypeptide that forms a protein of specific structure and function. Proteins, in turn, bring about an organism's observable phenotype. A given type of cell expresses only a subset of its genes. This is an essential feature in multicellular organisms. You'd be in trouble if the lens cells in your eyes started expressing the genes for hair proteins, which are normally expressed only in hair follicle cells. Gene expression is precisely regulated, which we'll explore in the next chapter, beginning with the simpler case of bacteria and continuing with eukaryotes. Concept check 17.5. What happens when one nucleotide pair is lost from the middle of the coding sequence of a gene? Make connections. Individuals heterozygous for the sickle cell allele are generally healthy but show phenotypic effects of the allele under some circumstances. See Figure 14.17. Explain in terms of gene expression. Draw it. The template strand of a gene includes the sequence 3-tact CATA C5. The sequence is mutated to 3-tact CATA C5. For both wild-type and mutant sequences, draw the double-stranded DNA, the resulting mRNA, and the amino acid sequence each encodes. What is the effect of the mutation on the amino acid sequence? What if? How could you use the CRISPR-Cas9 system to correct the mutation described in question 3? Based on the answer to question 3, would this be worth doing? For suggested answers, see Appendix A. Summary of key concepts. Go to Mastering Biology for assignments, the e-text, the study area, and dynamic study modules. To review key terms, go to the vocabulary self-quiz in the Mastering Biology e-text or study area, or go to goo.gl/zkjz9t. Concept 17.1 Genes specify proteins via transcription and translation, pp. 336. 342. Beadle and Tatum studies of mutant strains of Neurospora led to the one gene, one polypeptide hypothesis. During gene expression, the information encoded in genes is used to make specific polypeptide chains, enzymes and other proteins, or RNA molecules. Transcription is the synthesis of RNA complementary to a template strand of DNA. Translation is the synthesis of a polypeptide whose amino acid sequence is specified by the nucleotide sequence in messenger RNA, mRNA. Genetic information is encoded as a sequence of non-overlapping nucleotide triplets, or codons. A codon in mRNA either is translated into an amino acid, 61 of the 64 codons, or serves as a stop signal, 3 codons. Codons must be read in the correct reading frame. Describe the process of gene expression, by which a gene affects the phenotype of an organism. Concept 17.2 Transcription is the DNA-directed synthesis of RNA, a closer look, pp. 342. 344. RNA synthesis is catalyzed by RNA polymerase, which links together RNA nucleotides complementary to a DNA template strand. Transcription follows the same base pairing rules as DNA replication, except that in RNA, uracil, U, substitutes for thymine, T. DNA with transcription unit, RNA polymerase transcribing template, 3' to 5', strand of DNA. A promoter region on the DNA precedes the transcription unit. The 3 stages of transcription are initiation, elongation, and termination. A promoter sequence, often including a TATA box in eukaryotes, establishes where RNA synthesis is initiated. Transcription factors help eukaryotic RNA polymerase recognize promoter sequences, forming a transcription initiation complex. Termination differs in bacteria and eukaryotes. Compare the initiation of transcription in bacteria and eukaryotes. Concept 17.3 Eukaryotic cells modify RNA after transcription, pp. 345. 347. Eukaryotic mRNAs undergo RNA processing, which includes RNA splicing, the addition of a modified nucleotide cap to the end, and the addition of a poly-A tail to the end. The processed mRNA includes an untranslated region, UTR or UTR, at each end of the coding segment. Most eukaryotic genes are split into segments, they have introns interspersed among the exons, the regions included in the mRNA. In RNA splicing, introns are removed and exons joined. RNA splicing is typically carried out by spliceosomes, but in some cases, RNA alone catalyzes its own splicing. The properties of RNA allow some RNAs, called ribozymes, to act as catalysts. The presence of introns allows for alternative RNA splicing. Pre-mRNA with 5' cap and 3' poly-A tail at the ends and segments of introns and exons in between. Arrow indicates introns are spliced out to form mRNA. 17.2 to 18 Full alternative text. What function do the cap and the poly-A tail serve on a eukaryotic mRNA? Concept 17.4 Translation is the RNA-directed synthesis of a polypeptide, a closer look, pp. 347. 356. A cell translates an mRNA message into protein using transfer RNAs, tRNAs. After being bound to a specific amino acid by an amino acylderna synthetase, a tRNA lines up by its anticodon at the complementary codon on mRNA. A ribosome, made up of ribosomal RNAs, rRNAs, and proteins, facilitates this coupling with binding sites for mRNA and tRNA. Ribosomes coordinate the 3 stages of translation, initiation, elongation, and termination. The formation of peptide bonds between amino acids is catalyzed by rRNAs as tRNAs move through the A and P sites and exit through the E site. mRNA inside ribosome. Transfer RNA binds to mRNA codon and transfers a PHE to growing polypeptide chain. Other tRNAs preparing to bind to mRNA codons and transfer their amino acids. 17.2 to 19 Full alternative text. After translation, during protein processing, proteins may be modified by cleavage or by attachment of sugars, lipids, phosphates, or other chemical groups. Free ribosomes in the cytosol initiate synthesis of all proteins, but proteins with a signal peptide are synthesized on the ER. A gene can be transcribed by multiple RNA polymerases simultaneously. Also, a single mRNA molecule can be translated simultaneously by a number of ribosomes, forming a polyribosome. In bacteria, these processes are coupled, but in eukaryotes they are separated in space and time by the nuclear membrane. How do tRNAs function in the context of the ribosome during translation? Concept 17.5 Mutations of one or a few nucleotides can affect protein structure and function, pp. 357. 362. Small-scale mutations include point mutations, changes in one DNA nucleotide pair, which may lead to production of non-functional proteins. Nucleotide pair substitutions can cause missense or nonsense mutations. Nucleotide pair insertions or deletions may produce frameshift mutations. Spontaneous mutations can occur during DNA replication, recombination, or repair. Chemical and physical mutagens cause DNA damage that can alter genes. The CRISPR-Cas9 system is a powerful new gene editing technique with the potential to correct genetic mutations that cause disease. Significant technical and ethical questions have been raised about how this could or should be used. What will be the results of chemically modifying one nucleotide base of a gene? What role is played by DNA repair systems in the cell? Test your understanding. For more multiple choice questions, go to the practice test in the Mastering Biology e-text or study area, or go to goo.gl/GRUWRG. Levels 1 to 2, Remembering/Understanding. In eukaryotic cells, transcription cannot begin until the two DNA strands have completely separated and exposed the promoter. Several transcription factors have bound to the promoter. The 5 caps are removed from the mRNA. The DNA introns are removed from the template. Which of the following is true of a codon? It never codes for the same amino acid as another codon. It can code for more than one amino acid. It can be either in DNA or in RNA. It is the basic unit of protein structure. The anticodon of a particular tRNA molecule is complementary to the corresponding mRNA codon. Complementary to the corresponding triplet in rRNA. The part of tRNA that bonds to a specific amino acid. Catalytic, making the tRNA a ribozyme. Which of the following is true of RNA processing? Exons are cut out before mRNA leaves the nucleus. Nucleotides are added at both ends of the RNA. Ribozymes may function in the addition of a cap. RNA splicing adds a poly A tail to the mRNA. Which component is directly involved in translation? RNA polymerase. Ribosome. Spliceosome. DNA. Levels 3 to 4, applying/analyzing. Using. Figure 17.6, identify a sequence of nucleotides in the DNA template strand for an mRNA coding for the polypeptide sequence PHE prolis. UAKA. Gakukt. Trkia. AOQ. Which of the following mutations would be most likely to have a harmful effect on an organism? A deletion of 3 nucleotides near the middle of a gene. A single nucleotide deletion in the middle of an intron. A single nucleotide deletion near the end of the coding sequence. A single nucleotide insertion downstream of, and close to, the start of the coding sequence. Would the coupling of the processes shown in Figure 17.24 be found in a eukaryotic cell? Explain why or why not. Complete the following table. Type of RNA. Functions. Messenger RNA, mRNA. Transfer RNA, tRNA. In a ribosome, plays a structural role, as a ribozyme, plays a catalytic role, catalyzes peptide bond formation. Primary transcript. Small RNAs in the spliceosome. Levels 5 to 6, evaluating/creating. Evolution connection. Most amino acids are coded for by a set of similar codons, see Figure 17.6. Propose at least one evolutionary explanation to account for this pattern. Scientific inquiry. Knowing that the genetic code is almost universal, a scientist uses molecular biological methods to insert the human globin gene, shown in Figure 17.12, into bacterial cells, hoping the cells will express it and synthesize functional globin protein. Instead, the protein produced is non-functional and is found to contain many fewer amino acids than does globin made by a eukaryotic cell. Explain why. Write about a theme. Information evolution accounts for the unity and diversity of life, and the continuity of life is based on heritable information in the form of DNA. In a short essay, 100 to 150 words, discuss how the fidelity with which DNA is inherited is related to the processes of evolution. Review the discussion of proofreading and DNA repair in Concept 16.2. Synthesize your knowledge. Siamese cat. Ears, area around eyes, nose, and mouth, feet and tail are dark. Rest of body is light. Some mutations result in proteins that function well at one temperature but are non-functional at a different, usually higher, temperature. Siamese cats have such a temperature-sensitive mutation in a gene encoding an enzyme that makes dark pigment in the fur. The mutation results in the breed's distinctive point markings and lighter body color, see the photo. Using this information and what you learned in the chapter, explain the pattern of the cat's fur pigmentation. For selected answers, see Appendix A. Explore scientific papers with Science in the Classroom AAAS. How is green fluorescent protein, GFP, used as a marker for gene expression? Go to Lighting Up Life at www.scienceintheclassroom.org. Instructors, questions can be assigned in Mastering Biology.
Chapter 16 is about DNA replication. So DNA replication happens in the S phase or the synthesis phase of the cell cycle. If you remember from chapter 12, we looked at the cell cycle. And a cell is usually in interphase. For most of the time, like 90% of the cell's life is spent in the interphase. And the interphase in turn is divided into 3 phases, the G1, S, and the G2. So the cell is normally, the newly formed cell is always in the G1 phase. And then once it gets a signal to divide, either through mitosis or meiosis, depending on the cell type, the cell then enters the S phase or the synthesis phase. And this is where every chromosome in the cell duplicates, right? So it becomes double-stranded, which we call as sister chromatids, right? And then after that, the G2 phase, and then mitosis or meiosis happen, followed by the cytokinesis, and then you have 2 cells or 4 cells, depending on the type of division, and then the cycle begins all over again. So we will be focusing on DNA replication that happens during the S phase in this chapter. Now, I will not be going over the history of how James Watson and Crick came up with a model or proposed a model that described the DNA structure. You can always read from the textbook. It's just so the model or the structure of the DNA that Watson and Crick put together is based on information that they gathered from various scientific labs, from various scientists. And that's the DNA model that we know of. DNA is a double-stranded molecule that is twisted over itself into a helix. So it's a double-stranded or a double helix, right? So think of it as a rope ladder with rungs, and then that is twisted over itself. So that's the DNA structure that James Watson and Crick proposed in 1953. Okay. And the rungs are made of nitrogenous bases, which are of 4 types in DNA. It is adenine, thymine, guanine, and cytosine, or ATGC. Right? And later, another scientist called Chargaff studied extensively the composition or how much of each of these nitrogenous bases are present in various DNA samples that he collected from different species. And what he observed was that the amount of A was always equal to the amount of T or thymine, and then the amount of guanine was always equal to the amount of cytosine, right? And that's something that Watson and Crick also used. That is an important piece of information for them to say, okay, if these bases exist in equal amounts, like A is always equal to T and G is always equal to C, then probably they are paired together, because we know that DNA is a double-stranded molecule. Okay, so that's called as the Chargaff's rule. Okay, the amount of A is equal to amount of T, and the amount of G is equal to the amount of C. Okay, so that's an important piece of information that Watson and Crick gathered. So you are familiar with this structure. We went over this in chapter 5 in a lot of detail. We said every strand of DNA has a 3' end and a 5' end. And the 5' end has the phosphate group. And so this is always the closed end. You cannot add nucleotides to this end. Nucleotide is the building block, right? So this is one building block. This is the next building block. See how all these building blocks are put together to make a strand of DNA, right? Now on this end, which is called the 3' end, you have the OH in the sugar. That is, the 3 stands for the 3rd carbon. So if you start the numbering, of course, you don't see the carbons here. It's just to make sure this image is simple. They have eliminated that. But that number indicates a carbon there at each corner of this pentagon, except for this one that has an oxygen here. So 1, 2, 3. On the 3rd carbon, there is an OH group that combines with the hydrogen of the next nucleic acid. The next nitrogenous base, and that basically forms this bond. See how this building block or this nucleotide is connected to this nucleotide with the help of this bond here, right? So a similar bond like this one is going to form between this nucleotide and the next nucleotide that's going to be brought in here. And all this happens during DNA replication. So this is how it is built. So 3' end is the open end for the DNA strand. And you will have another strand like this parallel, or actually anti-parallel, meaning the second strand of the double-stranded DNA molecule will have the 5' end here, because this strand has the 3' end. And then for the second strand, the 3' end would be here, right? So that arrangement of the opposite direction of the 2 strands is called as the anti-parallel arrangement. Okay, so imagine this entire thing here. So you have these nitrogenous bases facing inward, and then you have the sugar phosphate backbone, which supports the entire structure, and you have these phosphodiester linkages between individual building blocks. Okay, so all this is part of the history that I was talking about. Another important scientist called Rosalind Franklin, she took an X-ray image of a DNA molecule, and that data confirmed that DNA is in fact a double helix. Okay, so this is the structure. I was talking about a double-stranded molecule, and you have one strand that goes in the direction of 5 prime 3 prime, the other strand in the direction of 3 prime 5 prime. So that's the anti-parallel arrangement. So they run in opposite directions. And you also notice that these bases pair up to a specific base. It's not a random pairing up of the bases, right? We already know that A always pairs up with a T with the help of 2 hydrogen bonds. So this dotted line you see here is a hydrogen bond. And then the G always pairs up with a C using 3 hydrogen bonds. Okay. Okay. And these are just different ways to represent the DNA molecule. And you will be seeing every one of these in this chapter. Okay. So that's the antiparallel arrangement of the DNA molecule. So again, we know that there are two groups of nitrogenous bases. One of them is purines, right? So that's purine. A and G are purines. Pyrimidines would be C and T, and the U of RNA, the uracil of RNA. So A and G are purines, C and T and U are pyrimidines. Okay. And this is just to show how the pairing between a purine and a pyrimidine is the same information that the X-ray image provided. But if you pair up a pyrimidine and a pyrimidine, see how narrow this DNA strand would be. That does not agree with the data provided in the X-ray image regarding the diameter of this or the width of the DNA strand. And if you had two purines together, it would be too wide. Two pyrimidines, too narrow. One purine, one pyrimidine, perfect match. Okay. So all these are different pieces of information, different data that Watson and Crick put together to build or propose a model, which we follow. Okay. So again, a refresher of the Chargaff's rule: A always equal to T, G is always equal to C. You don't have to worry about the structures. You don't have to memorize the structures you see here, but this is just to show the hydrogen bonding and why there are only two or there are three between these nucleotides. It's just their chemical nature that determines the number of hydrogen bonds that would form between these different bases or base pairs. Okay, so now we move on to the actual DNA replication process. And this process involves several enzymes or proteins. Most enzymes are proteins. So that's why it says many proteins. But it's actually several enzymes that help copy the information on one DNA molecule onto another DNA molecule. So it's just making a complement of itself, right? And that's what this DNA replication is all about. So by the end of this process, the cell would have reached the end of S phase, and by then you would have a chromosome making an identical copy of itself, which we call as sister chromatids in mitosis and meiosis, right? So it's the same base pairing rule that is followed here to build a new strand of DNA. Okay, so what happens is you have, this is the parental molecule. It's a double-stranded DNA molecule, right? So what happens in this process? Again, this process also was proposed by Watson and Crick. So what they proposed was one of the enzymes that helps replicate DNA or involved in DNA synthesis breaks these strands apart. Okay, so that's what they would look like. And each of these strands would now be used as a template. Okay, and then a complementary strand would be built here. Another complementary strand would be built here. So this is what you end up with at the end of DNA synthesis. So this is the very, very simplified version of what exactly happens to this DNA molecule during the S phase. This is what it happens. This becomes twice as much DNA, right? So by the time the cell enters G2, it would have twice the amount of DNA. Right? And you can see how each of these two newly made DNA molecules has one strand that belongs to the parental DNA and another strand that is newly synthesized, shown here in light blue. Okay? So this is what we will be following. Okay? So this model is also called as the semi-conservative model. That's because, like we saw in the earlier image, the daughter DNA strand has half or one strand that is the parental strand, and the other half or the other strand is the newly made strand. That's why it's called as a semi-conservative. Semi is half. To conserve is to have the parental DNA strand, right? So each daughter DNA molecule has one parent strand conserved and one newly made strand. Okay. Of course, there were other models that were proposed at the same time that Watson and Crick were working on their model of semi-conservative replication. So one was called the conservative, where an entirely new strand is built that is identical to this, but they couldn't really prove it through experiment. Same thing goes for the dispersive model, which is like you have the daughter strands that are a combination, random combination of the parental DNA. And this was also not proved experimentally. But Watson and Crick were able to prove that this is, in fact, the process of DNA replication. Okay, so again, as we go through this chapter, there are several differences in the DNA replication process between the eukaryotes and prokaryotes. I have them listed at the end of the chapter, but it happens at such great speed because of the enzymes, because there are enzymes present that make this process much faster. Of course, we will be following both of these processes, like what happens if it's a prokaryotic cell and what happens if it's a eukaryotic cell, and so on. So whether it is prokaryotic cell or eukaryotic cell, the replication process, or the point where the two strands are separated, remember that's the first thing that happens. The parental strands are separated, are broken apart. So that begins, that pulling the two strands apart begins at a very specific location on the DNA molecule. It's called the origin of replication. Now here you have the first difference between the prokaryotic and eukaryotic replication. The prokaryotes have a single origin of replication because all they have is one circular chromosome. So very little DNA to replicate, so they really don't need multiple origins of replication. So a single origin of replication in the prokaryotic DNA, and then you have hundreds of origins of replication in the eukaryotic DNA. That's because eukaryotes have multiple chromosomes. And also they have a lot more genetic material compared to a prokaryotic cell. So that's one difference right there. Okay, so this is what the prokaryotic chromosome looks like. It's a circular molecule, and see, this is the point. So it's basically a specific sequence of ATGCs that is identified as the origin of replication by a certain enzyme, and it starts opening the DNA, or by breaking the hydrogen bonds between these two strands. Remember, these two strands are held together by hydrogen bonds. So those bonds are broken by that enzyme, and that process begins at this location. And once it begins, see, there is a little bubble-like thing formed. That's called the replication bubble. And this grows bigger on both sides. See, you can see this double-headed arrow that indicates that this strand grows in this direction and this direction. So you have DNA being replicated in both directions, and eventually you will see how it's almost done here, and then by the end you will have twice the amount of DNA as you started off with. And here you see the replication process in eukaryotes. Each DNA molecule has multiple origins of replication, and again the same thing, those enzymes attach at that locations, and they start breaking the hydrogen bonds and pulling the two strands apart. And see how the replication bubble grows bigger and bigger until all of these bubbles merge, and then you have 1 and 2 molecules of DNA from one molecule of DNA. So this is what happens in the eukaryotic cells. Okay.
Replication of a DNA molecule begins with a specific sequence called an origin of replication. The two strands separate, forming a small replication bubble. DNA replication starts and then proceeds in both directions until the original DNA molecule is completely replicated. Each of the two daughter molecules consists of one old strand, shown in purple, and one new strand, shown in green. The DNA of a eukaryotic chromosome has multiple origins of replication. The multiple replication bubbles allow this very long molecule to be copied quite quickly, resulting in two daughter DNA molecules.
Okay, so this chapter, you have a few enzymes that you need to memorize, their names and the function. Okay, so everything is highlighted here, and at the end of this chapter, you will also see a list of enzymes and their corresponding functions in the DNA replication process. Okay, so at the end of each replication bubble, notice how at the origin of replication, the DNA strands are pulled apart, forming a little bubble, and that bubble grows bigger and bigger, and that's so. On each end of that bubble, you have a fork called as a replication fork, which is a Y-shaped region of the bubble. Okay, so the first enzyme that actually untwists the DNA strand and then separates the two strands at the origin of replication, those enzymes are called as helicases. So the enzyme that identifies the origin of replication and then starts untwisting the DNA and then pulling the two strands apart by breaking the hydrogen bonds. Now, once the hydrogen bonds are broken, usually the two strands tend to come back together and reform the hydrogen bonds. Now, you don't want that to happen because you want the two strands separate so that their information can be copied and a new strand can be made. Right? So what happens is, to prevent the two separated DNA strands from coming back together, you have another set of enzymes called as single strand binding proteins that bind to the single stranded DNA that has been separated just now by helicase, and that prevents them from reforming the hydrogen bonds. Okay, so you need that. So this is the sequence, right? Helicase comes in, pulls them apart. Single strand binding protein comes next, and then make sure it just clings onto the single strand DNA and prevents it from binding with the second strand. And then now, as helicase just pulls the two strands apart, sometimes what happens is the DNA kind of gets over twisted, and that puts a lot of strain on the DNA molecule, and it might even break and be destroyed. So to prevent that overwinding of DNA, you have another enzyme called topoisomerase. This binds to the DNA, or it just smooths the DNA so that it relieves the overwinding of the DNA. Okay? So this is the position of these three enzymes, right? So that's just part of the replication bubble. You will have a mirror image of this on the other side of this replication bubble, right? So that would be the replication fork, the Y-shaped part of the replication bubble. So you have the helicase untwisting and separating the the two strands, and then right behind helicase you have the single strand binding proteins that make sure that these two strands don't go back together. And then you have topoisomerase kind of going ahead of helicase, making sure this is not over twisted. Okay, so that's the position. And then, of course, you have the primase. We'll talk about the primase in just a minute. You can see how there is some RNA also here. Now, you might think, why do we have RNA when we're talking about the DNA replication? That is very, very important. That's because now the new strand is built by an enzyme called as DNA polymerase, right? Because we are building a polymer of nucleotides, right? Of DNA nucleotides, so it becomes a DNA. And the enzyme that helps build a DNA polymer is a DNA polymerase. Now, the DNA polymerase is the actual enzyme that makes the new DNA strand by copying the information from the parent or the template strand. But the problem is the DNA polymerase cannot start the DNA replication from scratch. Okay, so it needs an already existing 3' end. Okay, now nucleotides have a 3', 5', whatever, right? But it cannot start building or putting together nucleotides so you have a 5' and a 3' end. It cannot do that. All it can do is just add more nucleotides to the 3' end and just make the strand longer, right? So we have another enzyme called as primase that can do that, that can actually start putting together nucleotides right from scratch. So we use that, or the cells use the enzyme primase, but the problem with primase is that it is an RNA primase, meaning it puts together RNA nucleotides. Therefore, the result would be a short strand of RNA. Okay, but that's okay. RNA also has a 3' end and a 5' end. So we'll just use that for now. Okay, so the RNA primer is made using or with the help of the RNA primase. Okay, and this is usually about 5 to 10 nucleotides long. So now you have a 3' end. Then the DNA polymerase comes in, and then it's like, I've got this. And then it just pushes away the primer, RNA primase, and then takes that location and then starts adding nucleotides to the 3' end of the RNA primer. Okay. And here is another difference in the DNA synthesis of prokaryotes and eukaryotes. It's the speed at which the DNA is synthesized. The bacteria or the prokaryotic cells can assemble 500 nucleotides per second. And in human cells, or most of the eukaryotic cells, it's 10 times slower. Okay, so there are only 50 nucleotides put together per second. And the reason for that is in bacteria, the DNA polymerase does not proofread the newly made DNA. It just makes it and then just goes ahead. Whereas in the eukaryotic cells, the newly made DNA is checked for accuracy. It is proofread for accuracy from time to time. And that's why it takes much longer, 10 times longer. But that's okay. We end up with DNA molecules that are that don't have any mutations, which is a good thing, right? But this process of proofreading gets kind of slower as we age. And what happens is the DNA proofreading mechanism also is it's not that effective. And therefore we end up with some mutations accumulating later on in our life, which leads to all these conditions, especially cancers. Most of them appear much later in life. And that's because by then we have accumulated quite a few mutations because our DNA polymerase is kind of not that effective in proofreading. Okay? So because this is a polymerization or DNA polymer is being built. If you remember from chapter 5, any polymer is built using the dehydration reaction, right? So the nucleotides are brought in at the DNA replication site, and they are brought in as triphosphates. Now, a nucleotide is a nitrogenous base, such as adenine, guanine, whatever, cytosine, thymine, and one sugar and one phosphate, right? So that's one nucleotide. But then the nucleotides that are going to be assembled as a new strand are brought in as a triphosphate, okay? And as the DNA polymerase completes the dehydration reaction, it gets rid of the two additional phosphates, okay, like this. Okay, so this is the triphosphate, the incoming nucleotide, but you don't want this and this because all you need is this, and this, a nitrogenous base, sugar, and a phosphate. But then this is how these nucleotides exist. So they are brought here, and then the DNA polymerase gets rid of the two additional phosphates, and then see how this T is added to the 3' end of this strand, right? And so this strand is getting longer now. Okay. So one important thing to remember is that the DNA polymerase adds nucleotides only to a free 3' end, not to the 5' end. That has a significant impact, especially in eukaryotic DNA. Okay, we'll talk about that a little later, but let's focus on the DNA replication as such in one replication fork. Remember, we have hundreds of these replication forks in our cells. Right now, most of our cells are going through mitosis, and they are at some stage of mitosis, or they are maybe making, or they are in the S phase of the cell cycle, right? So here you have, this is the origin of replication, and you have the helicase that is working its way, right? See how this is working its way and then opening more and more of this DNA, and this bubble is only going to get bigger and bigger, right? And you notice that this strand of the parental strand is used as a template, and so is this strand, right? They are both used as templates. That's the whole point, right? And at the end, you will have one double-stranded molecule of DNA and a second double-stranded DNA molecule. Now, from the point of origin of replication, DNA replication happens on both sides, right? And you might see all these arrows pointing in different directions. That's because now here it's mentioned it's 5', so we will just assume that this is 5' of the parental DNA strand. So this newly made strand that is shown in light blue, it's growing in this direction. If it's growing in this direction, that means this has to be the 3' end, correct? And that's why it's growing in that direction, because we said that DNA grows only in the 3' direction, right? 5' to 3'. And that's our little primer, that little red dot there at the end of each arrow is the primer, okay, the RNA primer, which will be removed later. Okay, so once the primer is made, the 3' end is available, and now the DNA polymerase starts to build. And see how this arrow just follows the helicase, or the replication fork. So the replication fork is growing in this direction, and so is this strand, right? So that strand is called the leading strand. The strand of newly made DNA that follows in the same direction as the replication fork is called the leading strand. Okay. Now, what about this strand here? Now, why isn't this growing this way? This is the replication fork here, right? Remember, we said this is the 5' end of this template strand, and so this is the 3' end of the template strand, which means the daughter strand will have the 5' end here. And the 5' end cannot grow longer, right? We cannot add nucleotides here. We can add only at the 3' end. So what happens here is a small primer is laid, and then this starts to grow in the 3' end. This is the 3' end. Wherever you see the arrow, that's the 3' end. Okay, so again, another is laid here, another primer, and then this grows in the 3' end until it reaches this strand. And then yet another primer is laid here, and then this grows in the 3' end direction. And then now it has to stop, right? And so why is it made as separate strands? Why can't this be made continuously? Now see how you cannot go any further here. Unless you open more of this, the template strand is not going to be accessible, right? So as this replication fork grows bigger, then you have more template DNA available for copying. And at that point, another primer is laid here. And then the DNA strand is, so the next arrow will be here. The little strand will be here. And then another little strand. And then at the end, all of these strands will be glued together to make a continuous strand. And then this strand will be added to this strand as well. So you have one continuous strand of DNA, and also all these red parts will be removed, and those will be filled with the blue parts or the DNA. Now, the same thing happens on the other side of the replication fork. Now, we said that's 5' and 3'. So this strand will have the 3' here and the 5' here, right? So if this is the 3' for the parent strand, this is the 5' for the daughter strand. Again, the same thing. It cannot grow in this direction. Therefore, it grows away from the replication fork, and that's why it's called as the lagging strand. So these words do not indicate the speed at which they are made. They just indicate the direction. Leading strand is made in the same direction as the replication fork. The lagging strand is made away or in the opposite direction of the replication fork. Okay, and then here, because this is the 5' end, the daughter strand will have the 3' end here. So from this point of origin of replication, the strand can just follow the replication fork. So in this replication bubble, this and this would be the leading strands, and these two little pieces of the lagging strand, those are the lagging strands. Okay, of course, the lagging strand will be put together. All these fragments will be put together, and they will be joined with the leading strand. Okay, so these little pieces are also called as Okazaki fragments, okay, which will be glued together later on at the end of the DNA replication. And okay, so that's important. A leading and lagging strand, like the direction. Let's watch this video.
Let's now look at the process of DNA replication. replication more closely, starting at an origin of replication. The DNA opens up there to form a small bubble. Molecules of an enzyme called helicase attach to the DNA at the ends of the bubble and continue the unwinding of the double helix. We'll focus on one end of the bubble, on the Y-shaped region called a replication fork. The two strands would naturally tend to rewind, but are held apart by molecules of a single-strand binding protein. The synthesis of a new strand begins when an enzyme called primase attaches and synthesizes a short RNA strand that is complementary to one of the DNA strands. This piece of RNA is a primer. DNA polymerase then adds DNA nucleotides to the 3' end of the primer. It continues to lengthen the new DNA strand by adding nucleotides complementary to the template strand. Notice that DNA synthesis always proceeds in a 5' to 3' direction. The strand just made here is called the leading strand. For clarity, we'll keep the new double-stranded DNA untwisted.
Okay, so the lagging strand, like I said, is made as in little segments, and those fragments are called as the Okazaki fragments, which are then joined together or glued together with the help of another enzyme called DNA ligase. Okay, so that's yet another enzyme that is used in the process of DNA replication. Okay, so far we have seen enzymes such as RNA primase, helicase, topoisomerase, single-strand binding proteins, and DNA polymerase, and then finally now DNA ligase. Okay, so here you have how the DNA lagging strand is made, right? So you have these little primers. So because there are multiple fragments, you have that many primers also. So that would be the first strand, second, third, and then you would have the fourth one here. So as more and more DNA is untwisted and opened up, you have more and more of these the Okazaki fragments being made, right? Now let's watch the lagging strand being made.
The other new strand is called the lagging strand. Unlike the leading strand, the lagging strand cannot be made continuously because DNA polymerase can only add nucleotides at the 3' end. Instead, the lagging strand must be made of fragments that are linked together. Despite their names, the leading and lagging strands are actually made at the same time. For clarity, here we will just show the lagging strand being pieced together. We'll start with a brief overview of fragments being formed and linked. First, an RNA primer, shown here in red, forms the beginning of each fragment. The rest of each fragment is then synthesized from DNA nucleotides in a 5' to 3' direction. The resulting segments are called Okazaki fragments. Next, each RNA primer is replaced with DNA. Then the gaps are closed to form the lagging strand. Now let's look closely at how enzymes work together to assemble the lagging strand. First, the enzyme primase removes the single-strand binding proteins, shown here in blue, and makes an RNA primer to begin an Okazaki fragment. The enzyme DNA polymerase then adds the complementary DNA nucleotides to synthesize the rest of the fragment. The assembly process continues as primase makes new RNA primers and DNA polymerase adds DNA nucleotides to create more Okazaki fragments. After the fragments are made, another kind of DNA polymerase replaces each RNA primer with DNA. Next, the enzyme DNA ligase links the Okazaki fragments to form the lagging strand. Here you can see the simultaneous formation of the leading and lagging strands. A DNA polymerase assembles a continuous leading strand while primase, other DNA polymerases, and ligase all work together to make the lagging strand. The enzyme helicase continues to untwist the double helix, exposing more template strand DNA for replication.
Okay, so here you can see all the enzymes in action right here. Now you also notice that there are two types of DNA polymerases. You have DNA polymerase 3, you have DNA polymerase 1. Now the DNA polymerase 3 is the one that is responsible for building both the leading and the lagging strands. Okay, remember that, it's very important. DNA polymerase 3 builds both the leading and see how it's also building the Okazaki fragment, or basically the lagging strand. Right? DNA polymerase 1, however, comes later in the DNA replication process. It gets rid of all these red parts, which are the RNA, and then fills it up with DNA nucleotides. Right? So that's another important process that happens at the end. And so the DNA polymerase 1 is responsible for replacing all the RNA nucleotides with the DNA nucleotides. And once those are replaced, you have DNA ligase, which is kind of like the glue, and it joins all these individual fragments together. And also when you have, now the leading strand also has an RNA primer, right? That also has to be removed and filled with DNA nucleotides. So again, you have DNA polymerase 1 working here, and then DNA ligase joining those strands together. So this is a very useful table that you see here. It has the list of all the enzymes and what their function is. So a good one to kind of review quickly with regards to just the function of each of these enzymes. So this is just showing how the DNA is replicated. See how you have a double-stranded DNA here, another double-stranded DNA here, okay, from this single strand, this double strand, the parental DNA. So we also talked about how the DNA polymerase in eukaryotes has the capacity to proofread any mistakes made during the DNA synthesis process. So let's say there is an A on the template DNA. So obviously you would have to have a T on the newly made DNA strand. But let's say there was a G placed there by mistake. The DNA polymerase is the one that made the mistake, and that's why it goes back a few steps, a few nucleotides, checks for accuracy in base pairing, and only if it is accurate, it moves ahead and then continues with the DNA replication process. But if there's any mismatch, it is repaired then and there. And that's the reason why the eukaryotic DNA replication is a lot slower compared to the prokaryotic DNA replication. Okay? Now, the DNA mismatch or the DNA damage can happen due to other reasons as well, not just during DNA replication. They can happen at any time, like say when you're exposing your skin too much to the ultraviolet light. Okay, what happens, or X-rays, all these can damage DNA. Now, if this exposure to UV or X-rays is minimal, these can be fixed because of our DNA polymerase. It can repair those little damages. But then if it is prolonged exposure to these agents like UV or X-rays or cigarette smoke, or any chemicals like arsenic, all these are potential mutagens, or they damage the DNA. So if there is prolonged exposure to any of these agents, then it's really hard for the DNA polymerase to keep up with the damage. And that's when you start to notice those effects in the form of primarily cancers. Okay, so the DNA repair kit has 3 very important enzymes. The first one is, now whenever it sees a mismatch, the first thing it needs to do is get rid of that incorrect base pair, right? We said there's supposed to be a T, but there is a G. What do we do then? Then it has to be removed. The G has to be removed. So you have to make a cut in the DNA, and that happens with the help of an enzyme called as a nuclease. Nucleases are a class of enzymes that make cuts in the nucleic acids such as DNA and RNA. So if you're making a cut in the DNA, it's called a DNA nuclease, and otherwise RNA nuclease. So the nuclease cuts the DNA and removes the damaged part or the incorrect bases, and then DNA polymerase fills in the correct bases, and then finally ligase, which is always the glue, that reforms the bonds between these nucleotides. That's called the DNA ligase. Okay, so that's the process of repair, DNA repair. In this order, you have nuclease that removes the damage, DNA polymerase that fills in with the bases, and then finally ligase that glues this patch up. Okay. Another problem with the DNA replication, especially only in the eukaryotes, not in the prokaryotes. Eukaryotes have linear DNA, right? Prokaryotes have a circular chromosome or a circular DNA. So they don't have this problem, but the eukaryotes do. So what happens is the DNA gets shorter and shorter with every round of DNA replication. And why is that? Now, see how, okay, this is a newly made DNA. This is a newly made DNA. Right? So that's a lagging strand. Remember, look at all these little fragments that are being put together, the Okazaki fragments, and each fragment has a primer, right? The little red part. Okay, so the DNA polymerase 1 goes in and gets rid of it. Okay, it removes it and then fills in with the blue or the DNA nucleotides, and then it goes to the last primer in the last Okazaki fragment, and then it gets rid of this red part, right? Remember, that's the 5' end. So it gets rid of this entire thing. So now this becomes the 5' end. See how this becomes the 5' end, right? And you cannot add nucleotides here, right? The DNA polymerase 1 cannot do anything. It cannot add nucleotides here. So that is just left shorter than this template strand. Okay, so your daughter strand, one of the two daughter strands here on this side is shorter. And the same thing happens to this one on the other end. It gets shorter. Right? So the DNA is now shorter. So later on, when this becomes the template, see how in the second round of replication, when this is used as a template, it's shorter. And this one gets even shorter. So as more and more of these replications happen, rounds of replication happens, you have shorter and shorter DNA. So that is a big problem because your DNA has important genetic information. You don't want to lose that genetic information. What do we do then? So we cannot avoid it, but we can postpone the erosion of the genes at the ends of the DNA molecules with the help of special sequences, again, this is also DNA, but it's tightly packed DNA, which we call as telomeres. So here you have, telo is the end. So just like you have centromeres, which is at the center of the chromosome, you have telomeres, which are ends of the chromosome. So the chromosomal ends have these regions called as telomeres. Telomeres don't have any genes, functional genes. All they are like blank sheets of paper. So it's okay. You can just rip a blank sheet of paper. You're not losing any information. You rip more and more of those blank sheets of paper. And then eventually you come to a point where you have information, genetic information, or notes written in your book, right? Now, if you rip one of those papers, you lose important information, right? So, and the cell would become abnormal. And if you leave that cell that way as an abnormal cell, it's going to divide and produce two abnormal cells. So at that point, the cells just go through apoptosis. Okay, you don't want those kind of cells, abnormal cells, to hang around. So the telomeres basically delay the process of losing genetic information or genetic erosion. Okay. So those highlighted regions that you see here, those are the telomeres or the darkly stained region. Okay. Now, so what happens is as we age, basically, our telomeres get shorter and shorter, right? Because when the shortening will happen, the erosion will happen, but we are not letting our genes or important information being eroded. It's the telomeres that are eroded. And that's fine. We're not losing any important information. That's the reason we are able to function normally, right? But then we go through meiosis and we produce gametes much later in life, right? After puberty. By then, our telomeres would have shortened quite a bit, right? So when you form, when your germ cells or the reproductive cells form or go through meiosis and eventually form gametes, you don't want these chromosomes with short telomeres to be passed on to the next generation, right? So they would start with a very short telomere. So they would lose their telomere much faster than the parents did, right? So how can we fix this problem? Now, only in the germ cells, that is the cells of your reproductive organs that go through meiosis, those are called germ cells, they have an enzyme called telomerase. Okay, so this telomerase makes sure that the telomere is regenerated before meiosis happens. So you are passing along a full-length telomere to your offspring. Okay. So this telomerase must be present only in the germ cells. Let's say if it's present in our skin cells. Okay. That means our cells will no longer go through apoptosis, which is a characteristic of cancer cells. They do not ever go through cell death, right? You don't want that to happen in your somatic cells, right? You want the telomeres to erode, to get shorter. And then you also want normal apoptosis to happen. We talked about where apoptosis is supposed to happen, where apoptosis is not supposed to happen. So you want that process to happen normally. So you don't want any telomerase. Now, scientists have recently discovered that certain cancer cells actually have telomerase in them. Now, how does this telomerase come into the cancer cells? Now, all cells have all the genes. It's just that the telomerase making gene is shut off in all of your cells except for your germ cells. But for some reason, that gene is turned on in your normal cells, and that to cancer cells. And they start producing telomerase, and that's what keeps the cancer or the cancerous cells from just keep dividing over and over again. Okay. All right, so now in this section, the last section, we will be talking about how DNA is packaged. Now, this packaging is important, especially in the eukaryotes. Okay, this has nothing to do with the prokaryotes, because prokaryotes just have one chromosome that is duplicated into 2 chromosomes, and it's so easy to just sort those 2 chromosomes and the bacterial cell to divide into 2, right? But in the eukaryotes, you have multiple chromosomes, and during mitosis or meiosis, these chromosomes are moved back and forth, and when you're moving them, you don't want the DNA to break, the DNA to be damaged, right? So how do we move these? It's just like when you move from one house to the other, right? Or when you're traveling, you don't just take everything separately. You actually pack everything in a suitcase, in a box, right? And then you unpack it when you reach your destination, right? So it's the same thing. Here, the chromosomes are packed tightly, and that's why you're able to see chromosomes under the microscope when a cell is in mitosis or meiosis only. When the cell is in interphase, it's all loosely arranged. Why is that? That's because only if it's loosely arranged, the cell can actually access the genes and then use those genes or that genetic information to make proteins, right? So that is important. Just like you have all your things in the house unpacked, right? You don't have them all tightly packed in a box. You can't use them. So the same way, unless your genetic information or your chromosomes are loosely arranged or unpacked, you cannot access the genetic information in the genes. But when you are ready to move the chromosomes around, you want to pack them up. Now, how can we switch between this packed and unpacked state? Okay, that's because we have some special proteins. The eukaryotic cells have proteins called as histones that change or alternate between positive and negative charge. And why is that? DNA, remember, always has a negative charge. And the reason it always has a negative charge is because it has a phosphate group. So it always has a negative charge. That doesn't change. What changes is the charge on the histones. So as the result of the cell division, okay, or right before cell division, when the cell is in G1, remember, there is a signal that the cell receives, and that's what triggers the cell division process, right, or preparation to divide. So this is one of those. So that trigger in G1 causes a chemical change in the histones, which gives them a positive charge. So now you have DNA and histones, which makes up our chromatin, right, that have opposite charges. So it's really easy to pack all the DNA around these histone proteins into tightly packed chromatin, right? So this process happens in multiple steps. The first step, or the basic packaging unit, is called a nucleosome, which is basically a bead. It looks like a bead, and that bead is made up of a cluster of histone proteins plus DNA wrapped around that cluster twice. Okay, so that's called as the nucleosome. Okay, so that's these are the nucleosomes, and they have a diameter of 10 nanometers. They look like a string of pearls, and that is folded over itself to form a 30 nanometer fiber, which is then looped into several loops. Looks like a flower, right? So you have several such layers of these flowers, and that's called as a looped domain. Each one is a scaffold, and that loop domain is folded over itself, and that forms the chromatid, the sister chromatid. And this is what you see in metaphase, right? By the time the cell reaches or starts metaphase, this is what the chromosomes look like. They have these two copies, right? Remember, we said all this happens during DNA replication, right? And so by the beginning of metaphase or end of prophase, this is what the chromosomes would look like: a replicated chromosome with a diameter of 1400 nanometers. Okay, so know this sequence of packaging. Okay, so the tightly packed chromatin is called as heterochromatin. Okay, densely packed, highly condensed chromatin is called heterochromatin. So all our chromosomes or chromatin is in the form of heterochromatin during cell division, mitosis or meiosis. And there are some parts of our chromosomes that are always tightly packed, or they are always in the form of heterochromatin, and those would be the centromeres, telomeres. And the reason for that is the telomeres or centromeres don't have any genetic information that our cells can use, right? They have a specific function. They are, of course, chromatin, but they don't have any genes on them. Okay, telomeres, they postpone the erosion of genes. Centromeres, they are present for moving the chromosomes around, right? Remember, this is where the kinetochore microtubules attach to the chromosome, right? So the rest of the time, where the cell is not dividing, the chromatin is loosely arranged, so it can be used for protein synthesis, and that is called as euchromatin. Eu is good, or something that can be used. Okay, euchromatin is the loosely packed chromatin. So it's the histones that actually go through chemical modifications that gives them a positive charge, or they are neutral. And so based on what the histones are like at different times of the cell cycle, your DNA can be packaged, or your chromatin can be packaged or loosely arranged. Okay, so this is the table I was talking about with the difference between the prokaryotic, eukaryotic DNA replication. It is an important piece of information for this DNA replication chapter. Okay, please get back to me if you have any questions regarding this chapter. Okay, bye. Okay, so in this chapter, we will be exploring gene expression. Gene expression can result in several molecules. So when you say gene expression, it's basically the information on a gene is used to make some kind of a molecule. So right now, you have in this slide, you see gene expression can result either in an mRNA, which then goes on to make a protein or a polypeptide, which is then processed to a protein, or the gene expression can result in a tRNA or a transfer RNA. Also, gene expression could be could end in rRNA or ribosomal RNA, and gene expression can also lead to several other RNAs. So all of these are a result of gene expression. So gene expression is not always protein synthesis, but protein synthesis is just one of the results of gene expression. So the gene expression by which DNA information is used to make a protein happens in two steps. So this is what we will be exploring in this chapter, the gene expression that results in a protein molecule. And this process happens in two steps. Now, this is the same whether it is in prokaryotes or eukaryotes. It has to happen in two steps. And we did talk about this earlier in the semester when we said the information, the flow of information in a cell is always from DNA to mRNA, and then mRNA to a protein. And then the proteins are used as tools by the cell to perform all of the functions that the cells perform. So the first step is called the transcription. So basically, it's like copying the information from a gene, a specific gene, onto an mRNA. Okay? Just like we saw how a new strand of DNA is made using one DNA strand as the template, right? So this process of transcription is very similar to that. The only difference is we use the DNA as a template, and then the result is not a DNA strand, but it is an RNA. Okay, so the mRNA that is made using the information on a DNA template is through complementary base pairing. So that process is called as transcription. That is the first step. Now we have the information or the recipe we need to make a polypeptide on our mRNA, also known as messenger RNA, right? So the transcription results in a messenger RNA, which is just a copy of the genetic code or the recipe we need to make a polypeptide. So now the next step is translation, which is basically using the information on mRNA and then building a chain of amino acids called as polypeptide. So the information on mRNA corresponds to a specific sequence of amino acids. And this process of translation happens at the ribosomes. And where are the ribosomes? The ribosomes are in the cytoplasm. And then in chapter 6, we also said that ribosomes in eukaryotic cells can be at two different locations, right? Ribosomes can be either free floating in the cytoplasm, or they could also be attached to the endoplasmic reticulum, depending on where the protein is used. If it's used by the cell, then it is made in the cytoplasm by the ribosomes or at the ribosomes. And if the protein is secreted out of the cell, then it has to be made on the ER or the endoplasmic reticulum from where it is transported out of the cell. So that's the only difference. Otherwise, all the ribosomes in a eukaryotic cell are identical. They can just shift places between cytoplasm and endoplasmic reticulum based on what kind of a protein they make at that particular instance. So these are the two steps, right? Now let's take a look at the difference between these steps or the protein synthesis in a bacterial cell, which is prokaryotic, and a typical eukaryotic cell. Now, eukaryotic cells, they are like so diverse, right? You have protists such as amoeba, paramecium, algae. All of these are eukaryotic. Then you have plants, also eukaryotic. You have animals, also eukaryotic. Fungi, eukaryotic. So all of those, the list of eukaryotes that I just mentioned, they have this general process. This is very similar in all of the eukaryotes, whether it is a fungus, an animal, a plant, or a protist. And a prokaryotic cell, this is what the protein synthesis looks like. Okay, so what difference do you see here between these two? Obviously, one of the major differences is the location of the transcription and translation. See how in a prokaryotic cell, both of these steps happen in the cytoplasm because there's no nucleus in prokaryotic cells. So that's one difference between the eukaryotic and prokaryotic gene expression or protein synthesis. Whereas in the eukaryotes, you have the transcription happening in the nucleus because that's where your DNA is. That's where your genetic information is. So the transcription has to happen in the nucleus. And then it results in mRNA that is eventually transported out of the nucleus into the cytoplasm. And then you have the translation happening in the cytoplasm. Right? So that's one difference. Then the other difference is here in the prokaryotes, look at the mRNA that is made at the end of transcription and compare that to the mRNA that is made at the end of transcription. So different, right? So here you see these little patches of lighter regions. So those are eventually spliced out, right? And the mRNA that is transported out of the nucleus looks a lot different from what is originally made by the transcription process. So there is something called as RNA processing that happens in the eukaryotes. That's another big difference between the protein synthesis of prokaryotes and protein synthesis of eukaryotes. There is no RNA processing in prokaryotes, whereas in the eukaryotes, the lighter regions are removed, and these lighter regions are called as introns. And then, so this mRNA that is made at the end of transcription is called a pre-mRNA, also known as a primary transcript, and that is processed and made into the functional mRNA or the mature mRNA, which is then exported out into the cytoplasm where it is translated. So what are the things that happen during RNA processing? Okay, these lighter regions are removed. Those are called as introns. And then you can also see that there are these yellow regions that are attached on either ends of this mRNA. So now this is the functional mRNA, meaning this is the one that has the actual code that determines what sequence of amino acids is present in the polypeptide. Whereas here, there's no such processing at all. Whatever is made at the end of transcription is used in the translation process, and then a polypeptide is made. Okay, so that we just talked about the primary transcript, or the transcript that is made, the mRNA that is made at the end of transcription in eukaryotes. And so the central dogma is basically how the information flows in cells. So it always flows from DNA via RNA to a protein. Okay. So now let's talk about the mRNA and what exactly is the information that is on this mRNA. Okay, that's called the genetic code. The genetic code is just instructions as to how and what and how many amino acids and in what sequence they should be assembled. So there are 20 different amino acids, and there are only 4 nucleotide bases in DNA, right? So how do we correlate these two? That is the genetic code and the corresponding amino acids. Okay, so when your mRNA is made and it is eventually exported out of the nucleus, if it's a eukaryotic cell, if it's a prokaryotic cell, it just stays in the cytoplasm, and then the mRNA information is read in the form of triplet codes. So a triplet code is a series of non-overlapping 3 nucleotide words. And remember, we are talking about mRNA, so there is no T, instead there is a U, right? So these 3-lettered non-overlapping words are called as triplet codes or triplet codons, okay? And each of these triplet codons corresponds to an amino acid, okay? So like for example here, this is a very small, simple example. You have the DNA template. And so the template is identified by its direction. It always starts from 3' and goes in the 5' direction. That's the DNA template, always. And so this is used as a template, and it is transcribed to make the mRNA through complementary base pairing, right? So if this is used as the template, then if there is an A on the DNA, remember we're making an RNA molecule, so instead of a T, there is a U, which is the complement of A. Because there is a C, we put a G. A C, another G. Now this makes up one triplet codon, and this corresponds to a specific amino acid. Now same thing, the next one, A, so you have U, which then forms one word, and that word means a certain amino acid in the amino acid language. So this is basically like translating this word in one language into another word in a different language. That's why this is called as translation. And this process happens in the cytoplasm, whether it is eukaryotic or prokaryotic cells, right? So, and see how they are non-overlapping, right? So that's also called as the reading frame. It's called a reading frame when they are read as continuous non-overlapping groups of 3 codons or 3 bases. Okay. So again, remember how the template codon, strand is identified. It is always in the direction of 3', 5', and the RNA is built in the 5' to 3' direction, which is normal for any nucleotide because the 3' is always the open end, right? And that's the reason your template codon, the template strand is always in the 3', 5' direction, so that you can build an mRNA or a polynucleotide in the 5' to 3' direction. Okay, so we also said that this is the template strand, and so the other strand would be known as the coding strand. And why is this called the coding strand, and why isn't this called the coding strand? Because we're using this as the template, correct? But this, the non-template strand, is called the coding strand. Now notice how, or compare this strand that goes in 5' to 3' direction to your mRNA. Okay, what do you think? There's a T, and instead of that you have an E because this is an mRNA, and then you have G and G, you have a G and G, you have T, you have U, G. So you see how this code on your mRNA is almost the same as the one on this strand, except for the U's and the T's, right? What that means is this is the actual recipe we want to copy on our mRNA. That's why this is called as the coding strand. And if you use this coding strand as the template, you will not get this result. You will end up with something like this, right? Because this process of transcription happens through complementary base pairing. So imagine if you used this as the template strand. Try that in your book, okay? Just write down the complement of this. It would look somewhat like this, but that's not our goal. We want this information on our mRNA. So what we do is we use this as our template strand, and then what we end up with is this mRNA, which is the complement, and it looks exactly like this, except for the U's and the T's. Okay, so your non-template strand is called the coding strand, and your template strand is called the non-coding strand. Okay. So that's very important to remember about the template and the coding strand. Okay, so your coding strand can never be the template because you want that code copied as is. So these three-lettered non-overlapping words are called as triplet codes or triplet codons. Okay, and each of these triplet codons corresponds to an amino acid. Okay? So, like for example here, this is a very small, simple example. You have the DNA template, and so the template is identified by its direction. It always starts from 3' and goes in the 5' direction. That's the DNA template, always. And so this is used as a template, and it is transcribed to make the mRNA through complementary base pairing, right? So if this is used as the template, then if there is an A on the DNA, remember we're making an RNA molecule, so instead of a T, there is a U, which is the complement of A. Because there is a C, we put a G. A C, another G. Now this makes up one triplet codon. And this corresponds to a specific amino acid. Now, same thing, the next one, AAA, so you have UUU, which then forms one word, and that word means a certain amino acid in the amino acid language. So this is basically like translating this word in one language into another word in a different language. That's why this is called as translation. And this process happens in the cytoplasm, whether it is eukaryotic or prokaryotic cells, right? So, and see how they are non-overlapping. Right? So that's also called as the reading frame. It's called a reading frame when they are read as continuous non-overlapping groups of 3 codons or 3 bases. Okay. So again, remember how the template codon, strand is identified. It is always in the direction of 3', 5'. And the RNA is built in the 5' to 3' direction, which is normal for any nucleotide because the 3' is always the open end, right? And that's the reason your template codon, the template strand is always in the 3', 5' direction, so that you can build an mRNA or a polynucleotide in the 5' to 3' direction. Okay, so we also said that this is the template strand, and so the other strand would be known as the coding strand. And why is this called the coding strand, and why isn't this called the coding strand? Because we're using this as the template, correct? But this, the non-template strand, is called the coding strand. Now notice how, or compare this strand that goes in 5' to 3' direction to your mRNA. Okay, what do you think? There's a T, and instead of that, you have an E because this is an mRNA, and then you have G and G, you have a G and G, you have TTT, you have UUU, GG, GG. So you see how this code on your mRNA is almost the same as the one on this strand, except for the U's and the T's, right? What that means is this is the actual recipe we want to copy on our mRNA. That's why this is called as the coding strand. And if you use this coding strand as the template, you will not get this result. You will end up with something like this, right? Because this process of transcription happens through complementary base pairing. So imagine if you used this as the template strand. Try that in your book, okay? Just write down the complement of this. It would look somewhat like this, but that's not our goal. We want this information on our mRNA. So what we do is we use this as our template strand, and then what we end up with is this mRNA, which is the complement, and it looks exactly like this, except for the U's and the T's. Okay, so your non-template strand is called the coding strand, and your template strand is called the non-coding strand. Okay. So that's very important to remember about the template and the coding strand, okay? So your coding strand can never be the template because you want that code copied as is, okay? So you use the other strand as the template, so you end up making or copying the exact recipe onto your mRNA. Okay. And we also said how the orientation determines which one is the template for a certain gene, and it's always that strand for that gene. Okay, now moving on to mRNA. So what it looks like and how it is read, right? How it is interpreted, how that information on the mRNA is used to build a polypeptide. Okay, and so this, we just talked about how this non-template strand is called the coding strand and all that. So each codon specifies the amino acid, right? So that's what we will be looking at. So it's kind of like a dictionary, right? That translates one language to the other. So here, the 2 languages that we are dealing with is the language of nucleotides, which is ATGC or AUGC, because we're talking about mRNA. And the other language is the language of proteins, which is the amino acids, right? The amino acids are like the letters of the alphabet for proteins, to build a protein. And for the mRNA, it is the AUGC, which are the letters. So you're translating between 2 languages. Now, we said that the AUGC is read in the form of 3-letter words. Now, we have 4 letters in AUGC, correct? Now, how many unique 3-letter combinations can we make? We can make 64 unique combinations. So there are a total of 64 unique codons possible using the 4 letters A, U, G, C in our RNAs. Okay. Now, out of these 64 unique words possible using the 4 letters A, U, G, C, 61 actually code for amino acids, and then the remaining 3 are just stop signals. They do not code for any amino acids. Okay. And another thing is we have 64 words and they correspond to 20 amino acids. Now, how is that possible? That's not equal in numbers, right? You have far more triplet codons than the amino acids, about 3 times more triplet codons. So how is this even possible? How do we make it work? That's because our genetic code is redundant, meaning there could be several triplet codons that code for a certain amino acid, but it is never ambiguous. That is, no codon specifies for more than one amino acid. Let's say there is a specific codon, AUG, and if that AUG is encountered by your tRNA, which is basically the translator here, and it comes across AUG and it has to decide whether it needs to pick up methionine or serine, different amino acids, that's not a good situation, right? You want to make sure that the tRNA has no doubt whatsoever as to what amino acid corresponds to that genetic code. Okay, but let's say if there's AUG and then there's AAA and then there's UUU and they all code for one specific amino acid, that's fine. That's not a problem at all. So what we say is the genetic code is redundant or repetitive, but it is never ambiguous. Okay, and we also talked about reading frame, which is basically the non-overlapping groupings of 3 letters on the mRNA. Okay, so this is kind of like what we call as the dictionary, right? Which helps us figure out, okay, if there is an UCU on the mRNA, what does that mean in terms of amino acids? It means serine or SER. Now, again, you don't have to worry about this dictionary. If there are questions based on this, I will be providing this codes to you, and all you have to do is just figure out, okay, if I give you a sequence of amino acids, amino acids, you should be able to tell me what code it could possibly be. Or if I give you a reading frame on an mRNA, you should be able to build a polypeptide chain for me using this information that I will be providing you with. Okay, so now, remember what I said about the redundancy or repetition in the genetic code? See how you have UUU and UUC both coding for phenylalanine, which is fine. So if UUU is encountered on an mRNA, the tRNA just simply fetches phenylalanine. Then it goes to the next codon and it comes across UUC. So it's still the same, phenylalanine. And then let's say it goes to the next code and it is UCA. Definitely it's going to pick up serine, right? So that's how the code on an mRNA is interpreted. Letters of or words that are 3-letter long, okay? And these 3 letters correspond to a certain amino acid. But there's never ambiguity. Like say, if you come across AUU, this AUU appears only here. That is isoleucine. You will not find AUU corresponding to any other amino acid. So there's no ambiguity. The cell does not have to decide whether it needs to pick up this or this or this. There's always only one amino acid, okay, coding for maybe more than one triplet codons. That's fine. So you have a total of 61 such triplet codons that correspond to the various amino acids, which are 20 basically, and then you have the 3 remaining stop codons, which do not code for any amino acids. Notice how it just says stop. Okay, so usually these codes appear at the end of the mRNA. Sometimes if there is a mutation, these might appear a little earlier on the mRNA, and that leads to a protein that is much shorter than what it's supposed to be, so it's going to be non-functional, right? So that's one of the mutations. Okay, so this code that you see here, this dictionary that you see here is called as the universal genetic code, meaning whether translation is happening in a bacterial cell or a human cell or a plant cell, a fungus, any cell, this is always, so CUU always means leucine. CCU always means proline. UUU always means phenylalanine. AUG always means start, and it also means methionine. So this code does not change. Now, what that does mean? What does that mean? It means that you can express a gene of one species in another species. Like I can take a gene from a human cell and insert it in a bacterial cell and express in the bacterial cell. Why? Because the genetic code is still the same, whether it is used in the bacterial translation or in the human cell translation. Okay, so that's basically what the universal genetic code means. And scientists have explored this by expressing some of the human genes in other organisms to make some of the human cell products like growth hormone or insulin. All of these are made in other cells, like say yeast cells or bacterial cells. So you can harvest a whole lot of these molecules in a very short period of time. So commercially, this is one of the ways that insulin is produced. And the insulin that is produced this way has very few side effects because it's not a chemical that is made in the lab. It is a gene product that is made in a living cell. Right? So here also you see some examples. There is this pig that is expressing a jellyfish gene. So the jellyfish glow in the dark. So you just insert the gene from the jellyfish into the pig cells and it expresses in the pig cells. And here you have a firefly gene being expressed in a plant. And all that is because of the universal. Okay, so let's take a closer look at transcription, which is the first step of protein synthesis. So transcription uses DNA as the template, and the result or the product of transcription is mRNA or messenger RNA. And so we also talked about how this mRNA is different in prokaryotes and eukaryotes. mRNA is processed and such, whereas in prokaryotes, there's no such processing. And this mRNA is synthesized with the help of an enzyme called as RNA polymerase. Just like we had DNA polymerase that makes a molecule of DNA using the information on the template, we have RNA polymerase that makes the RNA strand or mRNA using the information on DNA. And so this RNA is complementary to the DNA template strand. Now, one thing about this RNA polymerase is that it does not need a primer, unlike DNA polymerase. So we saw in DNA synthesis how there was a primer created by the primase enzyme, which was later removed at the end of the DNA synthesis. But RNA polymerase does not have that need for a primer. It can build right from scratch. So this is kind of like the general process of how transcription happens. This is just to show where it begins and how it ends, and what this specific region where the RNA polymerase binds with the DNA and then starts pulling them apart, and at the same time using one of these strands, that is the 3' 5' end, so you can build the RNA in the 5' 3' direction. Right? See how, and also this RNA polymerase just keeps moving along the DNA strand as it copies the information on the template onto an mRNA transcript. And then once it encounters, so this particular area where the RNA polymerase binds with the DNA and recognizes that as the beginning of the genetic code or the gene, that region is called the promoter. The promoter region is where your RNA polymerase binds with the DNA. And then it starts moving along. And so the region that is actually copied onto mRNA is called the transcription unit. Okay? And once it encounters a region called as the terminator region, that's when this process of transcription is completed, and then you have an mRNA made. And see how the RNA polymerase falls apart once it encounters this region called as the terminator region. Okay, so this promoter region is very, very specific. And this is where the polymerase binds to the DNA. That's what we were talking about. And the region that is actually copied is called the transcription unit. And then it just separates at the terminator. So in bacteria, it is very, very straightforward. There is a terminator region, and that's about it. Just like you saw in the earlier picture, the RNA polymerase just separates. Whereas in the eukaryotes, it's a lot more complex, and that's what we'll be following here. So in the eukaryotic transcription, you have three stages. Initiation, which is basically binding of the RNA polymerase to the promoter region. And because we're talking about the eukaryotic transcription, there are several other factors also, or proteins, that are needed for the transcription to initiate or to begin. And then after that, there is the elongation, which is basically building an mRNA using complementary base pairing rule. And then you have termination, which is basically ending the transcription process. So in the eukaryotes, the initiation is not as simple as we saw in the earlier slide, where you had the RNA polymerase bind at the promoter region and then just move along the DNA and copy it. Here in the eukaryotes, within the promoter, there is a specific region where the RNA polymerase binds. So this is another big difference between the prokaryotic transcription and eukaryotic transcription. Within the promoter region, in the eukaryotic gene, there is something called as the TATA box. Okay, and this is where the initiation complex, okay, the transcription initiation complex binds. Now, so we're calling this a complex, which means there's not just one single molecule such as the RNA polymerase. There is more than just the RNA polymerase. So in the eukaryotes, you have a specific polymerase called RNA polymerase II that makes the mRNA, okay, that does the transcription. So in the prokaryotes, there's no such distinction. There's no separate RNA polymerases for, say, the mRNA, the tRNA, the rRNA, and so on. There's just one kind of RNA polymerase that builds all the different types of RNAs, whereas in the eukaryotes, you have a different RNA polymerase for each kind of RNAs made. mRNA uses RNA polymerase II. So RNA polymerase II, along with something called as transcription factors. Remember, we talked about transcription factors in chapter 11, that is cell communication. So when a hormone or a steroid binds with a receptor, that combination is called a transcription factor. And a gene to be expressed in a eukaryotic cell needs several such transcription factors along with RNA polymerase II. So together, all of these form something called as the transcription initiation complex, which binds with the DNA in the promoter region at a specific spot called as the TATA box. Now, this is called a TATA box because this section of the promoter has only T's and A's. And why is there only T's and A's? That's because, see how this RNA polymerase has to separate the DNA strands apart. So A's and T's share only 2 hydrogen bonds, and the G's and the C's share 3 hydrogen bonds each. So it's easier to separate the strands where there are more of these A's and T's because there are fewer hydrogen bonds to break. So that's why you have the specific region in the promoter that is made entirely of A's and T's. And that's where these transcription factors identify that region and bind at that region. And then you have the RNA polymerase to see how much more complex this is in the eukaryotes. Now, what this means is a gene is expressed. So for a gene to be expressed, you have to have that many signals coming from outside of the cell. So the gene expression is under a lot of control in the eukaryotes. You need several signals coming from outside, and each of these signals is a transcription factor. So eukaryotes, that's another big difference between the eukaryotes and the prokaryotic transcription. So that's the initiation. And then the elongation. So in the eukaryotes, you have about 40 nucleotides put together in the mRNA per second. And so a gene can be transcribed simultaneously by several RNA polymerases. So if you have one initiation complex made, and then it starts to move along the DNA, copying that information onto mRNA, you can have another transcription factor and RNA polymerase II forming another initiation complex at the same TATA box of the same gene, and it can make another copy of mRNA, and then as it moves ahead, you can have a third initiation complex, and so on. So it's endless. It just depends on how much of that signal is received by that cell. So RNA polymerase II forming another initiation complex at the same TATA box of the same gene, and it can make another copy of mRNA, and then as it moves ahead, you can have a third initiation complex, and so on. So it's endless. It just depends on how much of that signal is received by that cell. So that determines how many times a gene is transcribed and how many copies of mRNA are made. And again, same thing, the 3' end is the open end for this mRNA molecule that is being built. Okay. So that's kind of like a close-up of what happens as the mRNA is built. See how this is, it just follows the base pairing rule. There is an A on the DNA, there has to be a U on the mRNA, and so on. Okay. Now, another difference between the bacterial transcription and the eukaryotic transcription is how they are terminated. So in the bacteria, the polymerase, the moment it encounters the terminator region, it just falls off. And that's it. You have your mRNA ready for translation. In the eukaryotes, however, the RNA molecule goes even further beyond the terminator region, and then it transcribes something called as the polyadenylation signal. Okay, so that is basically a region beyond the terminator region. And why is this extra piece transcribed in the mRNA in eukaryotes? that just helps process the mRNA. Remember how at the beginning we saw some differences? We saw that in the eukaryotes, there was this yellow piece that was added at the beginning and at the end of the mRNA, right? So this additional piece that is transcribed at the end of the gene helps add those yellow pieces, right? So that's one more difference. Okay, so this polyadenylation signal is also transcribed onto the mRNA in the eukaryotes, but there's nothing like this happening in the prokaryotes. Then the next step after transcription in the eukaryotes is that we modify the mRNA, right? There is the primary transcript that is made, and that is modified in three ways. One is the 5' end of mRNA is modified. The 3' end is modified, so you have both ends being modified of the primary transcript. And then you have those lighter sections or the non-coding regions called as the introns that are cut out and removed. Okay, so this is how the mRNA is modified in the eukaryotes. So on the 5' end, there is a 5' cap. Now, what does this do? This helps attach the mRNA to the ribosome. Okay, and so this 5' cap is nothing but a GTP, guanosine or guanine triphosphates, kind of like your ATP. So that's added at the 5' as a 5' cap, which helps attach to the ribosomes during translation, and it also protects the mRNA from degradation. Okay, in the cytoplasm, there are several of these hydrolytic enzymes. Now, hydrolysis is to break down polymers. mRNA is a polynucleotide, right? It's a polymer. So these hydrolytic enzymes tend to break down the mRNA. We don't want that to happen. We want to use the information on mRNA to make a polypeptide chain. So the mRNA has to survive until it is translated. So by adding a 5' cap, you are prolonging the longevity of mRNA, right? And also it helps attach to the ribosome. Then the 3' end gets a poly A tail. Remember that little region that was transcribed in addition to the actual genetic code? Now at the 3' end, there is a long chain of adenines added. That's why it's called poly A. So a very long chain of adenines is added at the 3' end. And that is possible because of the polyadenylation signal that was added to the 3' end during transcription. Okay. And then the third way it is modified. So first one, the 5' cap. Second way that is modified is adding the poly A tail. And then finally, you have the splicing or removal of these non-coding regions shown as light orange color here, or the peach color regions here. So that's the guanine triphosphate, right, GTP, at the 5' end. And this was the little polyadenylation signal that was added beyond the stop region or the terminator region. And you have your poly A tail added at the 3' end because of this. If this is not transcribed, then this cannot be added, right? Okay, so 1, 2, and then splicing these out. 3 ways in which mRNA or the primary transcript is modified before it is exported into the cytoplasm. Okay, so those non-coding regions are called as introns. Okay, these are introns or intervening sequences. And all the other regions that actually code for amino acids are called as exons. So these are the regions that are expressed. That's why they're called as exons. And this is the code that is read in the form of triplet codons, and they are used to build amino acid sequences. So RNA splicing is a process that gets rid of the introns and just joins all the exons together, so that you have now a continuous stretch of mRNA that codes for the amino acid sequence. Okay, see how your mature mRNA or functional mRNA is so much shorter than your primary transcript. Okay, so all these regions are removed, and then your darker regions or the exons are joined together. to make a continuous coding segment. So this is the one that is actually translated. So the mRNA that is being translated will have a 5' cap, will have a poly A tail, will have a continuous coding segment, but will not have any introns. Now, none of these things happen in prokaryotic or bacterial transcription or gene expression. None of these three things that we just listed. All these happen only in the eukaryotes. So the complex that is responsible for splicing out or removing the introns, that complex is called as a spliceosome. So the spliceosome contains several proteins and little RNAs that actually function just like an enzyme. So you see all these little loops. So those small RNAs, they function just like a catalyst would do. So they just speed up the process of cutting this intron out. See how the intron is removed, and then you have a continuous stretch of exons. So this spliceosome contains several such catalytic or enzyme-like RNAs, or RNAs that have enzyme-like function. So those are called as ribozymes. These are nothing but catalytic RNA molecules that function just like an enzyme. Now, earlier we said enzymes are mostly proteins. That is true. There are a small group that are non-protein, which are basically RNA molecules. Right? Okay, so another important thing about the RNA splicing is called as alternative RNA splicing. What this means is you can use the same mRNA or the primary transcript, and you can splice it in different ways and create different mature mRNAs. And what that means is each of these different mature mRNAs will then go on to produce different or slightly different proteins. Okay. So what this means is a single gene can be responsible for producing more than one protein. And that's how eukaryotic cells have a lot more proteins than the number of genes they have in their cells. Okay? And that's because of alternative RNA splicing. Okay? So each of these different sections are called as domains in the protein. And each of these domains may be coded by a different combination of exons. So this is called as exon shuffling. See how you have each of these exons that make a different domain in this protein, or they can also make completely different proteins, right? So if you, let's say, make a cut here, and then you make a cut here and then join exon 1 and 2, that would be one combination. Or if you just make a cut here and lose this entire part, and then you make a cut here and then join exon 2 and 3, that would be another combination. Or you can simply remove these two introns and join all the 3 exons. That's another combination. So there are so many different ways in these in which these exons can be joined or shuffled. So that's called as alternative RNA splicing that results in multiple proteins from a single gene. Okay, now let's move on to the next part, which is the translation. Translation is basically the process in which the code on the mRNA is used as a template and a polypeptide is produced. Okay, and here we have a very, very important molecule that plays an extremely important role. That's called as the tRNA. Now the T stands for transfer, not translation. Okay, even though this RNA is used in the process of translation, the T here stands for transfer because it literally transfers the amino acids from a different location in the cytoplasm to the location where translation is happening, which is basically the ribosome, right? The surface of the ribosome. Okay. So let's take a look at the translation process, like a quick view of the translation process. I will be going over this process in a lot more detail, but this is how the ribosome is, and see it has two sections, the large and the small subunit, and between these two subunits, the mRNA is held and it is read 3 letters at a time. And this reading is done by the tRNA. The tRNA reads the code on the mRNA 3 letters at a time. That we talked about the triplet codon, and it reads with the help of something called as an anticodon. So it's kind of like the scanner or a barcode reader that we use in a supermarket, right? So when you kind of like put the barcode reader on the barcode, it identifies that as a certain price or a certain item. So it's the same thing. So this anticodon is kind of like the barcode reader, and the codon here on the mRNA is the barcode. So once the tRNA uses its anticodon and figures out, oh, okay, it says CGG, which means it has, or GGC rather, because it's always read from the 5' to 3' direction. Okay, so it's GGC, which means I have to fetch glycine. And so the tRNA is playing a very, very important role here. It figures out exactly what amino acid goes in the chain. Okay. Okay, so the anticodon is basically complementary to the mRNA, which basically is what your DNA is, right? Because your mRNA is complementary to your DNA, right? And your anticodon is also complementary to mRNA, which basically makes the anticodon the same as the information on DNA, except that your anticodon would have a U instead of a T, because anticodon is part of an mRNA molecule called a tRNA. So this is basically the structure of a tRNA. It looks kind of like a double-stranded molecule, but it is a single-stranded molecule, which all RNA molecules are. They are single-stranded. Now, the tRNA has two important regions. One is the amino acid attachment region. So this is the place that holds the amino acid and pulls it to the location where the translation is happening. And then the anticodon region, which is the barcode reader that we just talked about. So these are the two important regions, and see how these RNA molecules also have directionality. So the 3' end is where your amino acid is attached. Okay, so that would be 3', and this part of the anticodon would be 5', right? So you should know how to kind of like write the codon and also make sure you have the direction correctly. Okay. So the ribosomes, let's take a look at the initiation complex here for translation. So it starts with the ribosomes. So the ribosomes are the location, the physical location where this translation happens. And the ribosomes are usually made of two parts, the large and the small subunit. And ribosomes, like we talked about them a lot earlier in the semester, we said a ribosome is nothing but a combination of protein and ribosomal RNA or rRNA. Now, earlier the ribosomes were thought to be similar or identical in bacteria and eukaryotic cells, but apparently there is a lot of difference in the proteins that are present in the ribosomes of bacteria and the ribosomes of eukaryotic cells, and also the rRNA that is present in each of the ribosomes in bacteria and the eukaryotes. So that is a big breakthrough. That's because scientists then figured out a way to destroy the bacterial ribosomes, thereby getting rid of the bacterial infection using antibiotics that specifically target the bacterial ribosomes without harming the eukaryotic ribosomes. Let's say a person has a bacterial infection, right? And then the normal antibiotics just don't work on this person. So what are the options do we have? We can use another line of antibiotics that target the bacterial ribosomes and destroy them. And these ribosomes are extremely important. A cell without a ribosome cannot survive, because if there's no ribosomes, you cannot make proteins. And if there are no proteins, there's no way you can have a cell that can function, because it has no tools to work. Okay. So on a ribosome, there are these three very important locations, and they each have a significance. It's called the PAE site. Okay, the P site holds the tRNA that carries the polypeptide chain. That's why the letter P. The A site on the ribosome holds the tRNA that carries the next amino acid that goes in the polypeptide chain. And then the E site is where the tRNA exits the ribosome. So let's take a look at this. Okay, so there you go. On the large subunit of the ribosome, you have the P, A, and E sites, right? So the A site is where your the tRNA with the next amino acid enters. See how you have the tRNA? It has just read this barcode here using its anticodon, and then it fetched the amino acid that corresponds to this codon. And so now it is ready to add this to the chain over here. Okay, so it docks here at the A site. Okay, and then, but the next step would be to add this amino acid to this chain here, and then make a peptide bond between this amino acid here and this amino acid here. And remember, in chapter 5, we said the C terminus of the amino acid chain is the open end. So obviously this is the C terminus, this is the N terminus. So this is where your carboxyl group is, and that's where the next amino acid is added through a dehydration reaction, right? So once this is added to this bond between this polypeptide and this tRNA is broken. And the moment this bond is broken, this tRNA then moves to the E site, or the exit site from where it exits. Okay, and see how this is the carboxyl end. And then now, because this amino acid is added here, this would be the tRNA that would be carrying this polypeptide chain. And so it moves from here to the next groove, which is the P, or the amino acid or the polypeptide groove, right? And now you have your A groove vacant for the next amino acid to come in, right? The next tRNA with the next amino acid. And so the three stages of translation. First thing is initiation. elongation, termination. Just like we have these 3 exact stages in transcription, we also have them in translation. Okay, and of course there are protein factors such as some catalysts that help with these translation processes. And another very important thing is it needs energy, so it uses ATP. Remember, you are building a protein molecule, meaning it is an anabolic process, and anabolic processes need energy input. They are endergonic, right? So ATP is used, and there are 2 ATP molecules used for every amino acid added to the amino acid chain. Okay, so let's talk about the initiation of translation. The first thing is a start codon on the mRNA, which is always AUG. Codes, okay, so that is recognized by the tRNA. Now again, this AUG also means methionine in the amino acid language. So the tRNA so methionine or MET is always the first amino acid for every single protein that we know of, because AUG is not only the start codon, it is also a codon that codes for amino acid methionine. Okay, so first the ribosomal, the small ribosomal subunit binds with the mRNA at the 5' end, and then the initiator tRNA comes with the AUG, corresponding amino acid for AUG, which is methionine. Okay, and then there are a whole bunch of proteins that also called as the initiation factors that bring all these together. The 2 subunits, that is the large and the small subunit, and then the mRNA. Okay, see how you have your tRNA that codes for AUG. So the corresponding anticodon for the tRNA should be UAC, correct? And AUG codes for methionine, so that's always the first amino acid. And that comes and docks in the A groove, and then from there it moves to the P site, okay? And then you have your entire initiation complex ready, okay? The large, the small subunit, the mRNA, the tRNA with the first amino acid, okay? And it needs 1 GTP, which is basically like ATP. So some cells make GTP during cellular respiration, some cells make ATP. They both have the same amount of energy. Okay. So amino acids are added at the C terminus, and so the elongation occurs in 3 steps. The codon recognition. So first thing, the codon is read using the anticodon, and then a peptide bond formation between the next amino acid and the existing chain of amino acids. And then finally, translocation, that is moving of the tRNA into the P site, and then from the tRNA that was in the P site moves to the E site and then exits. Okay, and the translation always begins at the 5' end of mRNA, and then it goes towards the 3' end. Okay. So, of course, it's a lot faster. Okay, let's take a look at the building or the elongation step. So see how you have your, when we initiated this complex, we had the first amino acid, that is the methionine, which is already here. There have been 3 more amino acids added after that. So now you have a tRNA at the P site holding the growing polypeptide chain, and then you have your next amino acid being brought in by the tRNA, and the anticodon reads this codon. And so that's the first step, codon recognition, right? And then for this bond, the peptide bond to form between this new amino acid and the existing amino acid chain, there is 1 energy molecule used, that is 1 GTP used. And then the peptide bond is formed. And from there, you have your, so right now, this entire thing is moved here onto this tRNA, which is at the A site. But then we said the tRNA that holds the polypeptide chain must be present in the P site. So there's already this one. So this moves to the E site, and then this then moves to the P site. That's called the translocation. See how the tRNA that holds the polypeptide chain moves from the A site to the P site. That's called the translocation. And then this process also needs 1 molecule of GTP. So every time an amino acid is added to the chain, you have 2 energy molecules used, either 2 GTPs or 2 ATPs. And then this one exits, and now you have your A site available for the next tRNA and the next amino acid, and the cycle continues until the end of the transcription region, that is the mRNA, right? So this process of adding amino acids continues until a stop codon is encountered. So there are 3 different stop codons. So some genes have one kind of a stop codon, the other have the other kind. So it has to be one of the 3 stop codons that is present at the end of the transcription region. And so what this stop codon does is it tells, or it signals the end of the mRNA. And so a release factor just adds a molecule of water instead of an amino acid, and that is kind of like the end of translation. Okay. See how you have a release factor, okay, which basically adds a molecule of water to this, and that completes the process of building or translation, and the result is your polypeptide. Okay. And 2 GTPs are again added there for the addition of the last, or the release factor. And there you have your polypeptide chain, which is then folded into the, so what we built here is a primary structure. And from here, this is converted or folded into the secondary structure, and then the tertiary structure, sometimes a quaternary structure, and that's when the protein is actually functional. And see how once the polypeptide is released, you have this entire translation complex fall apart. Okay. So the polypeptide chains are modified, like I said, they are folded, twisted, braided, all of that, to give it a 3-dimensional shape that is unique to that protein. Okay. So this process is called as post-translational modification. Okay. Now here, remember earlier I said the proteins that are made by a cell may be used by the cell itself or may be transported out of the cell. Now how does the ribosome know whether this protein that it's making, or the polypeptide it is making or putting together, has to stay in the cytoplasm or has to be exported out, which means it has to be attached to the endoplasmic reticulum. Okay. So there is something called as a signal peptide. Okay. So the signal peptide indicates that this is a protein that has to be exported out of the cell, and that's when the polypeptide or the entire translation complex is moved and attached to the ER. Okay. So the signal peptide binds to something called as signal recognition particle, which basically drags the entire translation unit or complex. Okay, so here you have the signal recognition particle, and then this, so the gene itself had a code on the that was copied on the mRNA, which then created this, or was translated into this little part of the peptide called as the signal peptide, which is then recognized by the SRP, or the signal recognition particle, and it attaches to it, and then see how it's hauling this entire complex and then attaching it to a receptor. See, and then that allows the protein to enter into the lumen of the endoplasmic reticulum. Okay, and once this process starts and the polypeptide enters the lumen, see how the signal recognition particle has done its job. All it needed to do was to haul this entire thing and place it on the ER, and then help the protein get inside the lumen of the ER. Right? And then the protein synthesis is completed, and now this protein has to be exported out of the cell. So what happens is this part is pinched off as a vesicle, and then it goes and attaches itself to the Golgi, where it is processed into the functional protein, and then again pinched off as a vesicle, and then eventually taken out of the cell. Okay, so that's basically translation and the post-translational modification. So basically protein synthesis is done. Okay, we talked about how there are multiple mRNA molecules made in a cell. Sometimes there are multiple polypeptides made using the same mRNA molecule. So the mRNA molecule is translated several times. And for that, there are several ribosomes that attach to the same mRNA, making several copies of the polypeptide. Right? So that's another way to make multiple copies or molecules of that protein. So this can happen at the transcription level, where you have multiple mRNAs, which are then translated into multiple molecules or several molecules of protein, or you can just make one mRNA and then translate it multiple times and make several copies of the polypeptide. So at any given point, you see an mRNA being translated by several ribosomes at the same time. That structure is called as a polyribosome. Take a look. So this orange string that you see here, that is the mRNA. So there was this first ribosome that started to translate, and see how it has already finished translating, completed polypeptide, right? So this is a real image of the mRNA being translated in a bacterial cell by several ribosomes, which means that many copies of the polypeptide are made. So this structure would be called as a polyribosome. Okay. And another thing about bacterial cells is they don't have this mRNA modification, right? So whatever is transcribed is also translated. But in the eukaryotic cells, between transcription and translation, you have the RNA processing step, right? So what that means is the bacterial cells can start transcription, and as the mRNA is made, it starts to be translated. So here you have the DNA, and you have the RNA polymerase that just made this red colored line, which is the mRNA. And as the mRNA is made, you have the ribosomes attaching to that, several of them attaching to it. And even before the transcription is completed, translation begins. Right? See how you have just a part of the mRNA, it's not even complete, and you already have the ribosome attached to it and started to translate. Okay. So this is how bacterial cells make a lot of protein in a very short period of time. And that's why they replicate so quickly, because they can speed up the protein synthesis process, because they don't have the mRNA transcription, the post-transcription modification or the mRNA modification process. So they can make several hundreds of copies in a single day, or actually a single hour. So it's a lot faster compared to what we see in eukaryotes. Okay. So there is that concurrent transcription and translation happening in the bacterial cells. Okay, but so again here we see that difference, right? In eukaryotes, because of the separation in the location of transcription and translation, this is a beautiful image. This gives you an idea of what happens and where each of these processes happens, and in a little detail than you saw earlier at the beginning of this chapter, right? So that's transcription happening, and then you have your primary transcript, which is then spliced, and then see how there is a poly A tail, and of course there is a 5' cap, and then now that exits the nucleus, and that is attached at the 5' end to the ribosomes, and then you have your translation happening, and then you have your polypeptide chain being made. Let's watch this video now.
This student is cramming for her biology test with only a frosting-covered donut for lunch. Specialized cells in her pancreas respond to the increasing amount of sugar in her blood by releasing insulin, a small protein that regulates blood sugar levels. Let's go inside one of these cells to see how this protein is manufactured. The instructions for making insulin are coded by a segment of DNA in the nucleus. In transcription, an enzyme zips along the DNA, forming RNA, shown here in red. RNA nucleotides line up with their complementary DNA partners, transcribing the information in DNA into RNA. As the RNA grows, it is processed in several ways. First, a modified guanine nucleotide is added to the beginning as a cap. Also, segments of the RNA strand that do not actually code for the protein are removed, and the remaining segments of RNA are reconnected. Finally, extra adenine nucleotides are added to the end of the RNA strand, forming a tail. The completed messenger RNA, mRNA, now leaves the nucleus. The message in mRNA is translated into a protein in the cytoplasm. First, a transfer RNA, tRNA, arrives, carrying a specific amino acid. The small subunit of a ribosome attaches to the mRNA. Now, the large subunit of the ribosome attaches. A second tRNA docks, bringing another amino acid. The ribosome helps to form a covalent bond between the two amino acids. The mRNA shifts, and the first tRNA leaves. A new tRNA brings another amino acid. The ribosome helps to form a new bond, and the process is repeated. Notice that one end of a tRNA molecule has a set of 3 bases, called an anticodon, that pairs with complementary bases on the mRNA. The other end of the tRNA carries a specific amino acid. Different types of tRNAs carry different amino acids. In this way, the message in mRNA is translated into a specific sequence of amino acids. For proteins that will be secreted from the cell, like insulin, the ribosome docks on the rough ER, and the protein grows into the ER compartment. The new protein molecules are packaged in a vesicle that is transported to the Golgi apparatus, where many proteins are processed. However, insulin is packaged in a vesicle that leaves the Golgi and is then processed. Proteins secreted from the cell are shipped to the plasma membrane. Here, insulin is secreted from the pancreas and begins to regulate the student's rising blood sugar levels.
Okay, so that wraps up the protein synthesis process. So the next part, or the last part of this chapter, is about mutations, specifically point mutations. Point mutations are changes in just one nucleotide or one nucleotide pair in a gene. Now, mutations can happen at a larger scale where you have a piece of chromosome missing, or a piece of chromosome being duplicated, or a piece of chromosome cut and pasted, inverted. So all of these abnormalities can happen at a larger scale. But here we are talking about a change in the genetic code that involves just one nucleotide pair, and that's why this is called as point mutation. Now, you might think, okay, it's just one nucleotide pair, so why is this such a big deal? That's because that one single letter change could mean a completely different amino acid, or it could change a normal triplet codon into a stop codon. So there are so many things that can happen, or sometimes it may not mean anything. So that's called as a silent mutation. So let's take a look at those different types of mutations. So what we saw this earlier also when we were talking about sickle cell anemia. How now this is the normal genetic code that is changed or altered, and now we have a mutation. So there is just a change of one base pair, right? The rest is the same here, and the rest of the gene, everything is the same. It's just this one pair that got switched, right? It just got swapped. So what this means is your mRNA would be different, right? So that would be your template DNA strand, and then so your mRNA would have GUG here. So instead of GAG, you have GUG. What that means is it's going to bring in or insert a different amino acid in the polypeptide chain. And because this is a different amino acid, this results in a different pattern of folding of this polypeptide chain compared to this. What that means is your protein will have a different shape, which then results in this altered shape or a sickle shape in the red blood cells. This is the normal appearance of red blood cells. And this is what happens when you have such abnormally folded proteins packaged inside this red blood cell. Okay, so these mutations, the point mutations, are basically divided into two major categories. One is called the single nucleotide pair substitution. So there are substitution mutations. The other ones are insertions or deletions. Okay, so these are the two major categories, substitutions or insertions and deletions. Now, out of these two categories, the insertion and deletion are more dangerous. They have a higher probability of producing very, very abnormal proteins, versus substitutions. They're not as bad as the insertions and deletions. And why is okay? So this is all these examples of what is happening. This is the silent mutation. They both code, see how the resulting amino acid sequence is the same even though there is a mutation here. Okay, so that's a silent mutation. Now, the insertion and deletions are the worst. That's because when you have a certain number, let's say 9 nucleotides on the mRNA, which makes three-letter words, right? Let's say you insert an additional letter. Now, your grouping of three-letter words is messed up. Same thing happens when you lose one of those 9 letters. Again, the grouping is messed up, right? So insertions and deletions have a very significant and disastrous effect on the resulting protein than the substitution. So such mutations are called as frame shift mutation. What that means is the grouping is changed because you either add an additional nucleotide or remove one of the nucleotides, which causes a frame shift. Okay. So what exactly causes all these mutations? There are several factors, physical and chemical. Okay. Those are called as mutagens. So physical factors could be all the radiations, the X-rays, the gamma rays, the ultraviolet rays. All of these high energy radiations, they are the physical mutagens, and the chemical mutagens could be something like arsenic in the food. There are several carcinogenic chemicals that have the potential to change the polypeptide sequence by changing the nucleotide sequence on the gene. Okay. So here you have all the kinds of mutations. So these are the substitution mutations. You have the silent, missense, nonsense. See how this just stopped abruptly and prematurely. Here you have the insertion and deletion. So frame shift. So the entire reading frame has changed because of the addition of that extra nucleotide. And here the frame has again shifted, and then you have, instead of this is the actual code or the amino acid sequence, and see how this has changed. So starting from that point where one has been deleted or added, you have all the amino acids following that point, from that point onwards, completely altered. That has a big effect on the overall structure of protein. And here a stop codon is encountered prematurely, okay, or there is a missing codon. Okay. So these are much more deleterious or disastrous compared to the substitution. Of course, nonsense mutation is as bad as the frame shift mutations.